for reminding me about draft. इस कॉमन सेंस के ड्राफ्ट में फंसा होगा. हाय, गुड मॉर्निंग. अच्छा, बताओ आपका. थैंक यू. आपका वो बहुत सुंदर बनाया है जो मेरा बनाया है.
अभी इसके बाद
हाँ, जरूर से जरूर से. वो उसका ये इधर. All right. Actually, I should take picture. This is the last day. कल तो एग्ज़ाम है पर आज ज़्यादा हैप्पी गोइंग है. कल थोड़ा स्ट्रेस्ड होंगे लाइक. कहाँ चला गया मेरा फ़ोन. ये 2 मिनट बाकी है. आप भी इधर ही आ जाइए जिससे आप भी आ जाए. I should have taken the picture the first day जब पूरा क्लासरूम भरा हुआ था. So that I can boast कितने सारे students थे. But then I can say these were the real students, right, who stayed till the end. शुरू करते हैं.
So good morning to all. So we welcome you on the lecture number 28. So ma'am, Professor Kavita ma'am will start our lecture on the title Targeted Drug Delivery Strategy for Precision Medicines. So ma'am will start.
Thank you, Leknath. So when we started this course, we started with what is cancer, right? And from there we discussed traditional medicines, right? No, not traditional medicine, traditional therapies. So like chemotherapy, radiotherapy, and surgery. From there we went to precision medicine, right? So you find the target and then you attack cancer. Now we are going to go one step further. So even if you have a medicine, okay, which is targeted therapy, okay, even then, if you do not allow it to just go to the cancer, so if you give it systemically, right, it can go to every cell of our body, and although it is a targeted therapy, it can still hurt normal cells. So that's where targeted drug delivery comes into picture. This is called TDD, targeted drug delivery. Okay, so the idea is that you get the drug only to the important site or the cancer site. So the rest of the body is spared. So that's the whole idea. Okay, so first thing in precision therapy is, of course, identify the target. Okay, and once you identify the target, and how do you identify the target? You use different kinds of omics technique. Whenever you hear the word omic means global, organism wide or tissue wide or cell wide. Okay, so you're looking at all the things. So if you're looking at proteins, it is proteomics. If you're looking at genes, it is genomics. If you're looking at metabolites, it is metabolomics. If you're looking at all the RNAs, it is transcriptomics. So that gives you the target. But then if you give a therapy which goes everywhere in the body, it can still be toxic. So now we have to go one step further and deliver the drug only to the tumor site. So in that case, suppose even if you are giving chemotherapy, as long as it only goes to the tumor site, it is not as bad, right? So what are the limitations of conventional therapy? Non-specific distribution, it goes throughout the body, and it will have off-target effects. We don't want to destroy healthy cells, we don't want to attack healthy cells, but it will attack healthy cells, right? And low bioavailability, only a small fraction of the administered dose actually reaches the thing. The whole thing is circulating in the body, liver is cleaning it up, which it is supposed to do. Okay, so only small portion. Now we know kinase therapy Kinase inhibitor therapy is extremely effective, right? Like Gleevec, it is very, very effective, but it has numerous side effects. And see, some of them are listed here. So, huge toxicity in the body, although it is a targeted therapy. If you give Herceptin, again, many, many, many side effects. So, our next step is to get the therapy directly to the tumor site. Okay, so that comes under targeted drug delivery, TDD system. So the magic bullet concept is that targeted delivery system concentrate the medication in the tissues of interest while reducing the relative concentration in healthy tissues. So you don't want healthy tissues to get impacted. So there are two kinds of targeting, active targeting and passive targeting. Okay, so passive targeting is that you are not specifically targeting, but the drug itself gets accumulated in the tumor site. That would be passive. I'll tell you more about it. And then there is active targeting, that you're really trying very hard to get it to the tumor site only. Okay. So there is this process called EPR, and that is what is used for passive targeting. So what is EPR? EPR is enhanced permeability and retention. So cancer cells are dividing, and then they are going angiogenesis. So they are not very, they don't, their whole job is to keep doing it, keep doing it, keep doing it all the time, right? So the blood vessels they make, they are not very nicely made. They are very leaky. So what happens is that whenever you give a drug systemically, it goes, and because see it is broken, the blood vessels are inherently broken at tumor sites, so the drugs will just leak to the tumor. So they are basically concentrated there without any effort from our side. That is passive targeting. So whenever you give a drug, they tend to accumulate at the tumor site because of EPR, because, and that is due to leaky vasculature. See, it is all broken. Tumors also lack effective lymphatic drainage, so the particles are trapped inside. So that is in our favor, correct? That the drug is getting there. Okay, see, lymphatic drainage is very poor in tumor cells. So that is what it is shown here, that this is passive targeting. It goes and the drugs are mainly accumulated there. Okay. Now I want to say something else here in this slide. So you can also, passive targeting also includes nanoparticles and biocompatible polymers and liposomes. So what they are is that suppose you have a drug and you give it orally just like that. So it is possible that it will be just destroyed in the digestive system, or even if you give intravenously in the blood also, it has a half-life, very short half-life. So it may not stay for very long. So what you can do, you can just package them in many different kinds of nanoparticles. So nanoparticles, there are numerous nanoparticles. It's a very old field now. So you trap the drug inside the nanoparticle, but the nanoparticle does not have any targeting signal. So it's like a drug inside a suitcase, correct? So it is just floating everywhere in the body with no distinction. With EPR, of course, it will accumulate at the tumor site, but then because you have trapped it inside, it lasts longer. So that is also passive targeting. You are giving it to them and it is accumulating there, but you're not really making an effort to only deliver at the tumor site. So that too will be called passive targeting. However, if you take the nanoparticle and attach some kind of, ligand which directly binds to the receptor at the tumor site, then it will become active targeting. Do you see the difference? Package it in a suitcase without address, passive. With exact address, active. Make sense? That is the difference between it. Okay, so now we come to active targeting. So there are many, many different ways to actively target to tumor sites. So these are some of them are shown here. So let's start from antibodies, right? So antibodies are extremely specific. So if tumor cells are expressing some antigen and you make an antibody, and those antigens are tumor specific antigen, TSA, tumor specific antigen, the antibody will carry the cargo to the tumor site. Exactly. Right? It is writing the zip code, everything there. Okay? And it will go there, latch there. How do and then it will deliver the drug. So I want to ask you one question. First question, how do antibodies deliver the drug inside the tumor? First question. And second question is that what is antibody mediated toxicity? So first answer this thing. What happens in your opinion? So this is let's say this is a cell, right? This is the surface of the cell. And my watch is something tumor antigen. So the antibodies are coming, okay? Antibodies are coming and they're binding it like this, okay? Then what happens? Once the antibody binds the antigen, what is the next step? Very good. Endocytosis. It is taken inside and the drug is also released inside and some of them will go outside. That's one thing. What else happens when antibody binds here? So antibody is Y-shaped, right? So let me now make my hand like an antibody. So antigen is here. The Y-shaped thing is bound here and this is the other lower part of the Y thing. What happens next after that? What else can happen? So this part is recognized by NK cells and all these things. They will come, the antibodies bound, NK cells will come and attack and eat up the tumor cells, right? So that is yet another way of doing it, okay? So you obviously can add antibodies, right? Then you can actually, there's so many things I'm just thinking. You could make specific aptamers. Do you guys know what are aptamers? Aptamers could be DNA aptamers, it could be RNA aptamers. Look at that. They don't have this kind of double stranded structure, not at all. These are secondary, tertiary structures of DNA, RNA aptamers, which are, so you use a process called SELEX, SELEX, and trillions of molecules are made and then you screen for the things which will bind to your antigen or your protein of interest. So you are going for tumor specific antigen and then if you take aptamer and attach it to your drug, it will only go and bind that tumor cell, right? So that is active targeting. Professor Venkatesh, during his postdoc, worked on folate receptor. So folate receptors are highly expressed in cancer cells and he took folate compound. So folate will bind folate receptor. So he attached fluorescent molecule to folate. He attached drugs to folate. So then folate, even if you give it as an oral pill or injection, it will circulate and then it will concentrate at the tumor site because tumor site antibodies will bind folate, internalize it, drug is inside the tumor, right? Or if it is bound there, it will be fluorescent. The tumor will become fluorescent. Correct? So it is like it is a diagnostic tool also. So you can imagine numerous things. So all you have to do, you have to look at what is specifically expressed in tumor cells. You attach your drug to that specific thing. If it is a receptor, you attach the ligand. If there's a ligand, you make a receptor, like an antibody is a receptor kind of thing. Make sense? So there are numerous ways of actively targeting there. So that is a very neat way of reducing toxicity of a drug. Okay. So I'm going to show you many examples. So ligand mediated targeting, like peptides, aptamers, or small molecules like folic acid, right? And then I will come to all of these things. So now we are in the active targeting mode. So folic acid, aptamer, carbohydrates, peptide, antibodies, anything can be used. Mainly you put the ligand or the receptor and then the linker and your drug of interest. You can take a nanoparticle and you can decorate it with folate. Now that nanoparticle will only be taken by tumor cells. So many examples. So ligand receptor interaction. Active targeting involves attaching specific ligands to the surface of the drug carrier, and then it will bind to the receptors which are overexpressed in those cancer cells. Okay, ligands can be antibodies, it can be small molecules, it can be aptamers, it can be anything. Okay, Receptors could be HER2 receptor, EGFR, folate receptor, transferrin receptor. These are different kinds of receptor. And then, as you said, Venkatesh, increased cellular uptake via receptor endocytosis, correct? Okay, so this is a beautiful picture. So this is the targeting ligand, this is the linker, and then this is the drug payload. Anything you want to deliver, you can deliver cytotoxin. Okay, you can attach therapeutic agents, you can attach imaging agents, it will fluoresce. Okay, so you can treat any disease. Cancer is just one of them. Anything you can. So this is your payload. This is your this is the receptor at the cell surface. This is cell membrane, and this is the ligand for that binds the receptor. Okay, and then the whole thing goes inside. Okay, this whole thing goes inside. This is shown here, aptamer. Look at the structure of the aptamer. Okay, this was very popular in 1990s. So this Selleck's method was developed by, I can't remember his name, somebody at Harvard. He was an expert in that, and that was so popular at that time, Selleck's method. But nowadays it's not as popular. It's a old technique, but you can actually create any kind of DNA or RNA aptamer. The other Gyan course I taught in 2016, that's when I talked about aptamers. And then you can attach your drug here. Nanoparticle, see, nanoparticle decorated with aptamers. Then they are taken inside and the drug is released inside. So nanoparticles can be used either passively or actively. Passively, that means you don't decorate the nanoparticle. You just give it, it is floating around. It's like a suitcase. Your drug is inside, but it has no address. Okay, whenever it reaches the tumor cells, yeah, it will release there also, but there is no specificity. But if you put the address, it will only go to the tumor site. Okay, so here this nanoparticle has the ligands expressing. So then it only binds to the receptor of the tumor cells. But here it is passively doing because there is no decoration outside. Okay. Now I've been telling you about pH sensitive things, right? So today I will tell you about stimuli responsive release. So in addition to that, what you do, you can attach folate and other things, or you can just make something on the outside the drug. So you can attach the drug with something so that it is a pro-drug. Okay, but it becomes a real drug once that thing is cleaved off, the pro part is cleaved off. And the pro part is pH sensitive. So remember, tumors, they secrete lactic acid. So the tumor microenvironment is acidic. So now you make acid sensitive linker. So that's pH sensitive linker. Then thermosensitive. You heat the tumor from outside. So when the drug goes there, it is thermal sensitive and it will release at the tumor site. Or you can magnetically guide the drug to the tumor site. So let's look at it. So this is pH sensitive. When it reaches the cancer cells, the microenvironment is acidic. So it will be only released there and it will go in the tumor cells. But here in normal cells, even if they take it, they will throw it out because it's not going to get released in there. Go ahead. As you said, we will use acidic environment, but in our body there are many different sites where, like in stomach also there are acidic pH. So it's probably not given orally, it's given intravenously so that it is only in the bloodstream. You're right, because it will fall apart in the digestive system, and there it is extremely acidic. Tumor microenvironment is not as acidic as the stomach. Okay, so look at this thing. They make a nanoparticle which falls apart. So in the rest of the blood's pH is around 7.4, so it is totally stable. And then once it reaches the tumor site, it drops to 6.8. Stomach is 5. It's very acidic. So this difference is enough for the nanoparticle to fall apart and the drug is released to the tumor site. So that is pH sensitive. Okay, now let's look at thermosensitive. So you actually make a nanoparticle which is temperature sensitive nanoparticle. you give it, right, through intravenous injection. Now, because of the EPR effect, it will more likely to accumulate at the tumor site, correct? EPR is enhanced. Exactly. So then it accumulates there, and now you have to give a thermal source from outside. And then these nanoparticles will release the drug exactly at the tumor site. Okay, so that is thermosensitive. Okay, now the magnetic guidance is very interesting. So you make nanoparticles which are magnetic. You know, you may be using some kind of beads for purifications which are magnetic, right? So it's a very simple thing to make magnetic beads these days. So you make magnetic nanoparticles and then you use outside magnet to guide it to the tumor site so that they accumulate at the tumor site. That is where it gets really interesting. So you can use magnetic fields to trigger drug release, that it falls apart once it reaches there, or you can also guide them to the right place. Okay, so it can be used in both ways. People have made a sperm hybrid micromotor. So this is a sperm. It looks like a sperm and it has drug loading here. Okay, and then they make this whole thing as a magnetic thing so that they guide it to the tumor, and once it reaches there, it just shoots the drug in there. Okay, so that was stimuli responsive release. Now we come to nanocarrier tool bags. So nanocarriers are nanoparticles. Okay, so you guys, all of you have heard of nanoparticles, right? It's a pretty old field now. So there are many different kinds of nanoparticles. These are the oldest one, lipid-based, liposomes, solid lipid nanoparticles. So how did this idea come? The membrane, the cell membrane, it is made of phospholipids. So that's how the idea came, the first idea, that what happens is that the cell membranes are hydrophobic, right? They have this kind of structure, right? You know, there is this hydrophilic part here, then the two tails, and then it is bilayer. So the two layers react with other two layers, non-covalent interaction, and this is hydrophilic. So this is hydrophilic, water outside, water inside, right? So initial liposome was derived from that idea that you make small vesicles of lipid bilayer and you pack stuff in there, and this lipid bilayer will fuse with the cell membrane, throw the stuff in there, right? So that was the first one. Then people just used all sorts of nanoparticles. Okay, so I'm going to show you. So look at this nanoparticle here. So this is the outside and this is the inside, lipid bilayer like, right? You can see here. So this is hydrophilic, inside is hydrophobic. So the molecule goes in because the lipid layer is hydrophobic, it goes in there. Hydrophilic can also get in there, fluorescent dyes can get there, and outside you can attach active targeting agent. You can attach an aptamer or folate or anything you want to attach, or antibody or peptide which will recognize specific places. So you can actually make a nanoparticle for active targeting. So this is just to show you all different, people have been extremely creative here. So polymeric missile, reverse missile, all sorts of missiles. Is there anyone working on nanoparticles here at IIT Indore? Probably not, right? Yeah. It's not a very new field here. Faculty members. Okay. Yeah, so nanoparticle is still a very interesting field. Look at all of these different kinds of nanoparticles there. Okay. So magnetic nanoparticles and nucleic acid, and this is acid activable. So if you put it, this will be activated in the tumor microenvironment. Magnetic, okay, so many of them. And the whole, this is a dendrimer kind of structure. So this is conjugated drug all around it, right? So you want to make a nanoparticle which is made of something which is biodegradable so that it doesn't cause too much toxicity. But having said that, people make gold nanoparticle. because gold itself is used for therapy. I think in one of them it said, uh, or maybe it will come, it shows that gold actually is used for therapy. So you can see different kinds of things. So there is one very important thing about nanoparticle. Okay, so there is something called homotypic targeting. What that means, I will show you that once again. If the two cells are of same kind, same cell membrane, right, then they will tend to interact with each other. So what they do, they take the nanoparticle and put an artificial membrane to mimic inside membrane, and then it will be interacting with each other. That is called homotypic. So what they do, they take the cancer cell membrane to mimic it and put the nanoparticles around this thing. So they will go to the cancer cells, like attracts like. So cancer cell membrane carrying nanoparticles will reach the cancer cells. Can you tell me another cell line which can target to cancer cells? If you have to package some cargo to tumor cells, which cells will you use? Mesenchymal stem cell. They are very good. They are very good. Trojan horse. They will find the tumor cells and they will go there. Okay. So inorganic, yeah, this is here. Why use inorganic nanoparticles? Gold nanoparticles are biocompatible. They have unique optical properties which allows for photothermal therapy alongside drug delivery. These days, photothermal therapy is becoming very important. I have quite a few friends and collaborators who are working on photothermal therapy. Okay, so and similarly, you have other kinds of these things. If you use iron oxide, it can be used for MRI imaging. Okay, so the point here is that there are many kinds of nanoparticles, and again, if you decorate them with proper ligands, you can actively target them to the cancer cells. Okay, now guess what? Immunotherapy is given just like that, just like in this, but they can also be packaged in nanoparticles and deliver it. Okay, so PD1, PD-L1 inhibitors are antibodies, are directly delivered to the tumor microenvironment, packaged in the nanoparticles. And these nanoparticles are loaded with PD1, PD-L1 inhibitors, and you can heat it up so that they're only released at the tumor site. So, you know, PD1, PD-L1 therapy is so effective to start with. But if you add a nanoparticle to it, it's even better because then there is less toxicity to healthy cells. Okay, all right. So now it is released just next to the tumor site. So it reduces toxicity. You guys already know about antibody drug conjugate. I'll tell you briefly. So this is your antibody, and now you attach the drug of interest using a chemical linker. And you guys already know that antibody will bind to the tumor site, and then the drug will be released next to it or inside it. And if it is released next to it, the tumor cells which are not even binding the antibody, because the drug is in the vicinity, they will be killed. And this is called bystander effect. Bystander. Remember that, okay? So this is a very important thing. Suppose the tumor is heterogeneous, which it is, right? So some cells are expressing the receptor for this antibody. So this antibody binds the receptor. So some of them will be taken inside and drug will be released inside. And when the cells die, the drug will be thrown out. Or some antibody will bind and the drug will be released there. And then they will attack other cells which do not even express the receptor. But it is within the tumor, correct? So it will kill other tumor cells. And this is called bystander. Like they are bystanders, right? They're just standing there. They're not doing anything, but they will get killed, which is very good, right? Because these are tumor cells. So that's what it is shown, that you give the ADC, it is taken inside and the drug is released, and then it will inhibit and the cells will die. Same thing here, okay? So target site has HER2, antibody will recognize it. This is your drug and this will be released. Okay, so this is the actual drug. You know, Herceptin is so good without the toxin. just binding there activates this part, basically activates the immune system, and K cells will come eat up. And if you attach a bad drug to it, which is a microtubule inhibitor, that is really good. So this drug becomes very good. This drug is called Kadcyla. Okay. So this is what I was telling you, bystander killing effect. So ADC binds to receptor, endocytocyst, and the drug is released here. This drug, some of the drug will be released from the antibody, and then all the other cells which are close by will also die, although they don't have the receptor. So you are even targeting heterogeneous tumor that way, which are not even expressing. Okay. All right. Biomimetic carriers, nature's disguise. So homotypic targeting is binds to the same tumor type. So natural extracellular, so what happens, do you guys know what are exosomes? Have you heard of it? How many of you? Not all of you, but some of you, right? So exosomes are vesicles secreted by cells. So what cancer cells do, they secrete these small exosomes. These are like nothing but small vesicles, and inside they will put microRNA and oncogenic RNAs and things like that, and they will go to innocent cells around which are not cancerous and go inside and fuse there. And once they fuse there, those genes will be expressed and those cells will become cancerous. So now we can use the same trick and package our drugs in exosomes and give it to patients, and that will work very well. So what is the advantage of using vesicle? Because the immune system does not attack it, because it's the same thing which is there in the body, right? So immune system let it go, and you can actively target exosomes to the tumor also. Okay, and it also can cross blood-brain barrier. So look at this thing. You can make exosomes having different kinds of packaging, like different kinds of suitcase to package your material in. You can use mesenchymal stem cell membrane. Okay, now they're going to go to tumor site. You can use red blood cells. You can use platelets, outside part. You can use immune cells, cancer cells, and all of these things will take the cargo inside. Okay, so this is just cell membrane coating. This is to fool the immune system, and you can get all different. See, all of them are different kinds of cell membrane coating. Okay, so this is biomimetic exosomes. So you can say mesenchymal stem cell membrane. You put your stuff there, you load your drug RNA, give it to the patient, and it will work. All right. So scientists are very creative. Okay, gene delivery vehicles. So this is gene therapy. Okay, so so remember I was telling you about transfection and viral infection. So transfection will not work in humans, okay, because even if you work with cells, it is very inefficient. So there's no way it will work with humans. So whatever DNA, suppose somebody has cystic fibrosis, right? And you want to deliver the correct gene for cystic fibrosis. You cannot give the DNA, it will get degraded in no time. So you have to package it. So you have to make a virus, which is not bad for the human, but it goes to the target site and releases the DNA, and that DNA becomes part of the host chromosome, and then the gene is expressed there. So those are viral vectors, and there are many different kinds of viral vectors like lentivirus, adenovirus, this kind of thing. So the efficiency is very high. If you put it in lipid nanoparticles, it's very poor. It says moderate, but it's pretty poor. Immunogenicity, because it's virus, the immune system will get activated. This immune system is fooled by lipid nanoparticles because they look like just lipid bilayers kind of thing. You can put limited cargo size because virus has that much space in there. You cannot put huge things, but here you can make large nanoparticles, package a lot of this. Viral vectors are relatively expensive, but the beauty is that it is permanent gene replacement. So that is the way to go. These have I'm guessing nobody has worked with viruses like I asked you before. So these are extremely easy to work with. These are pretty old things now. Lentiviruses came out in 2003, so we have been using for 22 years. It's very old. Retrovirus I've been using since the very beginning, since it was discovered in 1990s. Okay, so the guy who discovered retrovirus, he was in David Baltimore's lab. He was a graduate student and he came to Bristol for postdoc, and that's when I started my postdoc. So we started almost at the same time. So his advisor got the Nobel Prize for retrovirus discovery, and the guy who discovered it was a postdoc next door. So I learned from him. So I've been using it. I was fortunate to be in touch with him. So I'm learning it from the guy who discovered it, who actually found it. Okay, so the point here is it's pretty old now, and it's very, very. So you take the vector in the genetic material, make the virus, it infects, and it becomes part of the host chromosome. So this is very, very good. You can directly give the viral particle, or you can make a cell line infected by viral particle, and you can give it to the host. And the beauty is that it can cross the blood-brain barrier also. So if you have to do something in the brain, you can treat it that way. Okay. Now, in addition to that, many of you know that, have you seen like people wear patches where insulin is pumped to them all the time? We wear watches which is keeping track of our heartbeat and all the other things. And people who have sleep apnea or atrial fibrillation, these smart watches can actually keep a track of everything. So these are these are other things. Okay, so insulin diabetes, artificial, we call it artificial pancreas. It's not a pancreas obviously, but it is constantly monitoring glucose, and it has insulin pump. Actually, this is very surprising. In 1999, when I joined Novartis, I needed insulin for my experiment, and it was all open lab. It was one company, like the money was company's money. So sharing was not like my money, your money, it's the same money. So I asked this guy, do you have some insulin? And he was joking, obviously. So he lifted his shirt and he said, I have an insulin pump. Do you want me to take it down? It was very scary to see that. So, but it was a point, I had never seen it, right? But the point is that it is that old. People were using it at that time. Okay, so there are microchip implants that release precise doses of osteoporosis medication or hormones programmed on program command or remote signal. Okay, there are also patches for nicotine or something. So that is becoming pretty common. Okay, so key challenges with targeted drug delivery: target heterogeneity. You target it for one thing, you can deliver it, but the tumor itself is very heterogeneous. So the cells you are targeting will die, but the ones which will not die will take over. So that's the problem. Immune system attacks. Oncolytic viruses are our favorite, but they don't survive, right? Toxicity. Okay, that nanoparticles can themselves be toxic. Synthesizing a simple drug is easy, but when you make nanoparticles, you can have batch to batch inconsistency. And if you make a complex nanoparticle, it can be tricky to synthesize. And high production costs can limit accessibility to these nanoparticles. Okay. So future look is that we use AI in career design. So predictive modeling, material scaling, personalized optimization. Our 30th lecture is all about AI. Okay, so you will learn a lot about what AI is doing in that area. Okay, sounds good. Okay, so conclusion is that we have to be precise, safe, and effective. So, okay, then I have put two slides which have nothing to do with targeted drug delivery, but it has to do with our discussion, right? So remember we were discussing if somebody is diagnosed with liver cancer, you suggested let's open it up, do the biopsy. I said no, it's not a great idea, right? So Then I found this thing, and it will make us feel very good about it, that liquid biopsy can detect the biomarkers in many, many, many different cancers. Okay, so we don't have to worry about opening up a patient and looking at it. So what happens is that these tumor cells shed DNA, tumor DNA in the blood, and you just take out the blood and you screen for it. Okay, so for lung cancer, it is extremely good, very effective. Colorectal, very good. Breast, prostate, ovarian, brain, even early stage solid tumors. So you can do liquid biopsy. And when somebody has uh, cancer of the digestive system, even taking the stool can be very effective. So for bladder cancer, urine is the best way, no invasive surgery. Lung cancer, blood plasma. Brain tumors, cerebrospinal fluid. Okay, head and neck cancer, plasma saliva. Okay, GI cancers, plasma stool. So we don't have to open up the person at all. We can just directly do the biopsy, analysis that way. So finding out what's wrong is much easier than what we thought. Okay, all right, that finishes up the first lecture. We have plenty of time. We can start the second lecture. Okay. Any questions? Leknath, any questions? This was more your cup of tea, little bit, right? So 29th lecture is a very interesting topic, and that is polypharmacology. Do you guys know what is the meaning of polypharmacology? Anyone? And how does it differ from polypharmacy? Anyone? If you hear the word polypharmacology, what would you guess? Poly means many. Pharmacology means pharmacology, you know. So what many? Very good, right? You are using many drugs. But polypharmacy is using many drugs. Polypharmacology is using one drug which is hitting many targets. That's the difference. Okay. So, for the longest period of time, the key word was magic bullet. That means you have one bullet and it hits the right target. That is the magic bullet. Okay. But now it is the magic shotgun. Okay. So cancer is such a complex disease. If you just kill one protein, it may not be enough. So we have to find all the bad guys and kill all the bad guys without touching any of the good guys. So that is polypharmacology. Is it making sense? So this is what it is showing, that you are hitting many things at once. This one drug is targeting tumor growth, drug resistance, and metastasis. No cell phones, sir. See, I teach a lot. I can detect within a second. Like you are doing like this or that, I know exactly where your eyes are, okay? So I teach 600 students class, and I can detect who is touching their cell phone. So it's easy for me to see here, very easy, okay? I don't even bring my own cell phone. I mean, here I brought it for picture purposes, but I never use cell phone. I never take it to my class. Okay, so polypharmacology is use of a single drug that targets multiple molecular targets to treat a single disease or multiple disease. Okay, so this approach contrasts with the traditional one drug, one target. For the longest time, when I was a chemist, you guys know, so when I became a chemical biologist, that time the whole world was going magic bullet, magic bullet, magic bullet, one target, one drug, one target, one drug. It doesn't work that way. The diseases are very complex. If you kill one guy, suppose there is a, just think of Steve Jobs, right? There was only one Steve Jobs, but when he passed away, iPhones are still coming because there is always a replacement. So cancer is a living system. You replace one, you target one protein, they will make some other protein, which will compensate sometimes even better than the previous one. Okay, or they will hire two, three people to compensate for that one person. So basically, the whole rewiring occurs. Okay, so multi-targeting, a single drug molecule is designed to bind and modulate multiple biological targets to achieve a more effective and potentially less toxic. So you can argue, why not we take a combination of drugs? Why do we have to make one drug? Of course, it will be easy to pop up one pill versus 10 pills at a time. But other than that, what could be other advantage of one drug? Less toxicity, and also there could be drug more specificity within the dirtiness, right? And also you are reducing drug interaction. One time, uh, I know of someone, so he had some serious complications, right? So he had to be given many antibiotics at once, and it was just very time sensitive. So I was there to pick up the medicine for this person. And so I went to the pharmacy. So the doctors called the pharmacy, and you have to pick the medicine from the pharmacy, right? So when the pharmacist was giving the drug, the system started sounding. This was the first time I heard this warning signal, like the police siren kind of thing was going on. So why the system was saying you cannot give these drugs in combination, because if you give this drug in combination, the system, it was 10 years, 2013, so 12 years ago, right? So AI was pretty primitive compared to today. But then, even that time, the AI said, you cannot give this drug combination, this is too toxic. So, but the doctor had prescribed, so I had to pick it up. So I had to sign on the behalf. If there's any toxicity, I'm responsible. Okay, because the pharmacist would not give, because the system was saying you cannot take these many drugs in combination. So polypharmacology means that you actually do whole FDA, whole clinical trial with that one drug and see how it affects the patients in large, like thousands of patients, right? So contrast with polypharmacy. Polypharmacy is different from polypharmacology is different from polypharmacy, which involves a patient taking multiple individual drugs. Okay, so so polypharmacology, this is the molecule, and it will hit target 1, target 2, target 3. These are all bad targets, but it will not inhibit any good target. So you have to select it against good targets. It should not hit any good target. And then if you take combination, you have slight improvement, but with this one drug, huge improvement. You guys know what is synergy? Can anyone explain to me what is synergy? Lekna, do you know what is synergy? Synergistic molecule? Yeah, that is, but what is synergy? Yeah, go ahead. So if this is like 50 effect, and this is 50, that is the definition of synergy. It is more than A plus B. So if A is 100, B is 100, it is not 200, it is 700. So it's more than additive effect, that is synergistic effect. That is what polypharmacology is aiming to do. Highly synergistic. Okay, and adverse effects are none in an ideal situation. So what is the advantage? Improved efficacy, fewer drug interaction because you're not taking combination, simple dosing. You would be very surprised. Remember, she's not here, Shivani, today. Okay, but Shivani was saying it is just really hard to take drugs, like, you cannot take lifelong drugs for something. Thyroxine, right? I'm pretty good in this thing, but I've heard compliance is really bad among patients. I know in my extended family, people will not take, because I keep telling them these are supplements. I mean, thyroxine is not a supplement, but there are a lot of supplements we take, like vitamins and calcium and all those kind of things. I said, what is the problem in taking supplements? Why do you have to leave it? You just keep on taking it. Like you eat food, you just take the medicine. and they don't have any side effect. So, but the truth is that there is a strong compliance issue worldwide. If you have to take more drugs, somebody is forgetting this drug versus that drug, or okay, छोड़ देते हैं आज कल ले लेंगे. But it is not good. Some drugs you can leave once in a while, other drugs you cannot. You leave one dose, you go back 1 week because the blood levels go down and it stops working for 1 week till the blood levels come back again. So simple dosing, reduced cost of course, and drug resistance. When you attack some problem through multiple ways, the cancer will not get time to develop a resistance. Okay, so challenges, of course, more polypharmacology, more potential of side effects. Okay, to find a drug which hits all the bad guys and not touch the good guys, it's hard. Okay, as safety risk, and also FDA hates to approve a dirty drug. If it is a clean drug, FDA can approve it. The more dirty the drug is, the more problem it has to get it approved, right? So that is also another problem. So you want to avoid any good targets, you have to make it promiscuous for all the bad guys here. Okay, so one old thinking was one drug, one target. Combination therapy we already talked about, and this is polypharmacology, one drug hitting many things at once. So what is drug combination? Right? Drug combination is that you are using more than one drug, and we discussed that in cancer. Remember BRAF and MEK inhibitors together, or EGFR and MET inhibitors. If you inhibit EGFR receptor signaling, it upregulates MET as a resistance mechanism. So if you give both, you kill them, right? So this is all still going on. This is HIV combination therapy, which use three antiviral drugs, three HIV and three retroviral drugs at once. Okay, the best. So this is still going on, and this will still go on. This is not going to go away. Combination therapy will keep going on, especially for complex diseases. Even right when I was telling cancer patients are given anti-angiogenic therapy plus PD-L1 therapy, and that is extremely effective. Okay, but today I'm going to tell you about molecules which can do both things. One thing, but capable of doing anti-angiogenesis and anti-proliferation. Okay, so this is just comparing combination therapy with polypharmacology. This is cocktail of drugs, many drugs like BRAF inhibitor, MEK inhibitor, multiple chemical entities, the risk of drug-drug interaction, complex pharmacokinetics. One drug has a short half-life, other drug has a long half-life, which one to take first, which one to take later. Sometimes you are supposed to take drug A at 6 a.m. but drug B at 6 p.m. and maybe you have to take drug A twice a day, drug B once a day. So it's pretty complex regimen. Okay, and higher pill burden, polypharmacology, one drug, many targets, single chemical entity. Because you have one drug, one molecule, low manufacturing cost. Okay, so this is the paradigm shift. So when I started my postdoc, the whole world was going after magic bullet, that one bullet hits one target in the cell. Now we are going for magic shotgun. It is hitting all the bad guys at once, and that is how we can overcome resistance. Okay, so this is the guy who gave the term Paul Ehrlich magic bullet. He's a Nobel laureate. Okay, he was a Nobel laureate. If he gave it, I don't know about when he coined it. He may be still around because magic bullet came in 1990s or 2000. So it's a pretty new word. Okay, and now we use magic drug. This is single target, this is multi-target. Okay, you guys already know about this thing. So for schizophrenia, there is a drug. Okay, and that is so surprising. So there are so many drugs. There are so many people who are suffering from that. Have you heard of any person who has that, even in from distance? No. So one of my student, who was my PhD student, developed it. He was the smartest guy. I have never seen smarter kid than him. 10, 15 years after he graduated from my lab, now he has those symptoms. People who are super smart, and if the energy is not directed towards the right direction, they may have some propensity to develop it. I don't know. But now there are drugs which inhibits many targets, and it is very effective for this disease. Okay, so you guys already know what is. So I still included this picture. This is really good. We have published many papers where we have used indole ring and modified it all around it. So our collaborator Bits Pilani, he made those compounds and we just tested them. But the beauty is that, look at the indole ring. Many of you are chemists here. So look at the indole ring. So if you substitute second position, fifth position, this will become anti-asthmatic drug. If you substitute fourth position here, it will become cardiovascular. Just see, just by changing the modification, you can target different kinds of things. So now if you have to do polypharmacology, you know exactly what to do, right? So why single target drug fails? Because so suppose I am very important, and if I leave, somebody else will replace me, right, as a teacher. If there is a CEO of a company who is extremely important, Bill Gates left Microsoft, it is doing extremely well, right? The CEO is someone else. So the cells are living. You remove one target, they will overexpress something else. So compensation. Okay, so solution is that you attack multiple things. So the point is to generate precisely dirty drugs. You are making dirty drugs, but right amount of dirtiness. Okay. So if target receptor is A, B, C, but not target D, E, F. Okay. So synergistic is more than 1 plus 1. Okay. So this is the formula. So if you check it out, then you can, have you ever worked with a synergistic, like you add two drugs and they have a synergistic impact? Have you done that? You will. If you use drug in combination against cancer cells, there will be drug combination which will be highly synergistic. Okay. So how do you do polypharmacology? How do you do it? Many ways to do it. But one way is repurposing of drug. How many of you have heard of repurposing of drug? What does that mean? Like one drug is already approved for a particular disease, and now you are using the same drug to target to treat another disease. That is very good. Repurposing. So repurposing became such a key word or buzzword 10 years ago. So the most important example of this is metformin. So how many of you have heard of metformin? And this is for diabetic, right? What is the commercial name here? Because my uncle was telling me the commercial name for metformin, and I said, I don't know. What is it called here? Is it called metformin? You guys are scientists, so you know metformin, but the commercial name is something else for metformin in India. And if you hear that word, all of you will recognize that name. But the point here is that this is one drug which is like a supplement, which is good for you. Okay, this is given to diabetes patient. It is also given to pre-diabetic patients who have a chance of developing diabetes, right? So metformin, what is the side effect of metformin? It is anti-cancer. How good? How good? You are pre-diabetic, so you are not going to develop diabetes, hopefully, if you keep control at your sugar level, but it is anti-cancer. It's really awesome, right? So what is the advantage of repurposing a drug? If you start a drug from the scratch, it's a very long process, like preclinical, clinical 1, clinical 2, clinical 3, but metformin has already passed everything for diabetes. So now you have to check the cancer, so you just go right up here at the final stage and see that. So it cuts the cost tremendously. Keep in mind, when a drug is developed, whether anywhere in the world, right, the drug is very expensive initially. Why? It is not the actual synthesis of the molecule. That may take 1000 rupees altogether, or even 500 rupees just to make the molecule. Correct? but it is the discovery process of 10 years which led to that. So the company has to recoup all that cost, and that is why the drug is very, very expensive initially. Okay, so but if you repurpose, you save tremendous amount of money. Okay, so polypharmacology. So I'm going to tell you many things. So polypharmacology is, it hits many kinds of different drugs. I have to tell you a joke, right? So I was making these presentations tomorrow. I had made it before I came here, but I reworked on it. So I don't have any of the AI programs, so I use the free AI programs. So ChatGPT doesn't let you make more than few figures, right? So it was funny. Yesterday was making figures after figures after figures, and it wasn't stopping me. So I thought maybe on Christmas Day they've opened it infinite. Which is, very few figures are there, but for fun I made like 20 figures and didn't stop me. So I said two of the one things happened. I'm the only one working on a Christmas day on ChatGPT. So it's allow me, or it is a Christmas present. And they said, whoever. After 2025, it said, okay, finally stop it. You have hit your limit for today. But it allowed me many more figures than a normal day. But this is one of those figures which I made yesterday. Okay. You can always find out a AI figure because it will have spelling mistake. Okay, invariably spelling mistake. So combination therapy, network, I will come to all of these things instead of telling you here. So polypharmacology can be achieved in many different ways, and these are some of the ways. Okay, so okay, uh, just one second, something is wrong here. Just one second. Yeah, something is wrong here. I accidentally put the wrong thing here. There were many of them here. Where I will go home and I'll correct it when I before I send it to you guys. Let's stop here. There are many ways to achieve polypharmacology. I spent 7 hours doing it. It is not that. Somehow I put the wrong slide here. Okay, we'll stop here. We'll look at the strategies after tea break. Okay, all right, sounds good. 2 मिनट के लिए बंद कर दीजिए. शुरू हो गया? अब कर रहे हैं, ठीक है, हाँ ठीक है. Okay, let's start. So, so now I'm going to tell you few ways by which people are able to make drugs which can attack multiple targets. So sometimes what you do, you just have simple one drug and it just is a magic drug. So when Gleevec was discovered, the truth is that for Gleevec, so Gleevec was developed by Novartis and it was developed against BCR-ABL. And when this drug was marketed, right? So that time nobody knew that it is such a magical drug in the sense that they had made it against BCR-ABL, but it also inhibited c-KIT and FLT3 receptors, which are tyrosine kinases. And they are also upregulated in CML. And Gleevec is against CML disease, chronic myeloid leukemia. So it just happened to be the magical drug which was hitting all the right targets. Okay, so later on people made a drug which was against pure BCR-ABL and that wasn't as effective. So the dirtier drug Gleevec was more effective, and that is when people realize you have to hit more than one target. So Imatinib or Gleevec hits BCR-ABL KIT PDGFR, although it was developed against BCR-ABL alone. But later on, when people realized, yeah, hitting more than one target is really good, now we intentionally make drug which can hit more than one target. Okay, and sorafenib is one of them. So again, the pros are it is simple dosing, lower cost, but then you have to choose your targets very efficiently. Okay, so one of the ways to make a drug which will hit multiple target is to do network-based polypharmacology. So you are not going after one target, you are going after one whole pathway or network. So suppose, so this is not the best picture here, but what it is showing here is that this is EGFR receptor, this is MAPK pathway, this is mTOR pathway, NF-kappa B pathway. So they are all connected. Signaling pathways are not linear, like they are going like this, but they're also going like this. They're going in network. So suppose you know that in this patient, this pathway is regulated, whole RAS pathway is upregulated because of KRAS. You make an inhibitor for KRAS mutant, but you also target downstream along with that. So you can actually, or suppose two things are talking three things, so you can make a drug which will hit the main nodes, okay, critical hubs. If you hit those, then you have a better chance of killing the disease. Okay, so that's what it is that you can, you know, these protein-protein interaction networks, you can, I mean, people have been making it for decades now. They were not very good before, but now they are very good, because what happens is that all of us, right, all of us worldwide, when we do gene chip analysis or RNA-seq or different kinds of network analysis, now everything is, journals will say you have to submit the raw data, right? So basically everybody has access to all the data and they can analyze the data. So it's becoming a big database of all the data. So now AI is also becoming smart. The richer the database, more things you can analyze, correct? So protein-protein interaction networks, gene regulatory networks, they are becoming more and more sophisticated. So you can say, okay, this person, we have to shut down this whole pathway here. So you can use proper combination. So a very simple example is simultaneous inhibition of PI3K, AKT, mTOR signaling. So you guys don't know what they are. So what happens is that, look at this thing. This is receptor tyrosine kinase. So signal goes here. It goes to PI3K. From PI3K, it goes to AKT. From AKT, it goes to many, including mTOR. So instead of inhibiting only PI3K or only AKT or only mTOR, what if you make a drug and you inhibit all three? So no signal can leak out. You are just inhibiting completely. So this is a drug, vertical pathway. This is a vertical pathway. You are hitting all three. You make a compound which hits all three. Yesterday I spent a lot of time so that I could find a figure where they show the whole molecule like this, and they say this part inhibits PI3K, this part inhibits mTOR, this part inhibits AKT. I could not find, because I was hoping for some color imaging, like red is AKT, one molecule. So suppose it is one molecule, this part is hitting this part of the kinase, that I could not find it because it may not be that simple, or I was just not able to search it properly. But the point here is that you can make a molecule which can hit all three of them. But here is the challenge. The more complicated the molecule, the more side effect it will have, because it may hit some other targets also, right? So, but the idea is that you can vertically inhibit the thing, like all three of them, or single molecule dual inhibitors, like you have actually one molecule, but it is hitting several of them. Drug combination is not polypharmacology, and PROTACs can be used along with small molecule. So you can make a PROTAC. What is a PROTAC? Can you tell me? Targeted degradation. So you have one part which will identify protein of interest, and one part will identify E3 ligase, right? So this molecule which identifies protein of interest can actually bind to more than one protein and degrade more than one protein. That would be polypharmacology, right? So so you can, so old paradigm was one drug, one target, one drug, one target, one disease. Now multi-target, you are hitting the whole network of proteins which are involved in that, and for one. Now there is another way to do that, and that is called fragment-based and scaffold hopping approaches. So what you do is that, how many of you know of fragment-based chemistry when you make molecules? Some idea? Can you try to explain to us? Let me explain to. So fragment-based, what is fragment-based drug discovery?
Are you talking about retrosynthesis?
No, not retrosynthesis. Fragment-based drug discovery.
Like we have already online available drug repositories.
Yeah, yeah.
Then it helps us to develop different fragments. By combining them fragments, we finally build one or two molecules.
Exactly. So what you can do is that, so you have many different fragments, like many different chemicals, right? So these are all this, like say, different kinds of fragment A to Z. Let's say 1 to 100, and here you have other fragments 101 to 200. You can combine them in every possible combination, two fragments, right? So you will develop a whole new bunch of compounds. That is, in very simple words, that is fragment-based discovery. Then what happens, you say, okay, compound number 12 here in fragment 1 binds AKT, compound number 22 binds mTOR. You combine them together. Can you inhibit both kinases together? Is it making sense? So, and also, see, the beauty of fragment-based drug discovery is that suppose you have to make, suppose you have 100 fragments here, 100 fragments here. Now you just have to clip them together. That is much easier. And you think about it, all permutation, you will make thousands of compounds, right? But if you have to build each and every compound, how many syntheses that would be? Here you have made large scale 100 compounds, large scale 100 compounds. And you can build thousands of compounds just by clicking it. You can even use click chemistry there, right? So the point here is that fragment-based drug discovery is a very powerful way for building up libraries of compound. So that's what it says, fragment-based, and I will tell you what is the meaning of scaffold hopping approaches. So this is just a very simplified version. You take fragment A, fragment C, and you couple them together, and you can make large molecules. This is actually a drug here. So this is fragment A, this is fragment B. This is FDA approved drug. Okay, so this is fragment B, and you are linking them together, and this hybrid molecule targets CSF1 receptor. Okay, now let me tell you what is scaffold hopping. So this is a natural compound, orones. Okay, now they have seen, instead of oxygen here, if you replace it with NH here, this is better. So if you do this, there is higher chemical stability. Now instead of N, you also introduce an N here. Okay, so it's imidazole-like structure. So this one, you gain all these properties. If you change orons to these things, here you are putting sulfur here. So this is scaffold hopping. The basic structure is the still 5-membered ring, but just by replacing with some heteroatoms, you are changing, you are making them highly stable, higher cell permeability, higher oral bioavailability, better pharmacokinetic properties, less target promiscuity and off-target interactions. So that is called scaffold hopping. Okay, so now how do you do the fragment-based drug discovery? So let's say these are the two fragments, A and B, right? So you can attach them by a linker. So A is there, B is there, there's a distance between them, that is linker, right? That is linker-based. Or you can fuse them together. So A is still there, B is still there, but there is no linker, they are fused together. Or you can merge them together, okay? Like Ardhanarishwar, okay, like half of this, half of that, or whatever, right? So it's that. So that is what it is. So this is linker, this is fused, and this is merged. So you can make all sorts of compounds, or you can make a compound which is not a merged, fused, or linker, but that compound itself hits many targets. Okay, so here is an example. See, here I'm just trying to find out which one is linker based and which one is. So this one is linker based. This is one part, this is one part, this is the proline like structure is the link. It's not proline like, but this is the guy which is the linker here. In this, dopamine and this pharmacophore are attached to form this compound. Okay, here is yet another compound, two pieces. I have some better examples here. Sunitinib, imatinib. Imatinib is actually not a, it was not developed as that compound, it just turned out to be that. But look at these compounds here, okay? This is just a linker, this is a fused drug here, okay? This is merged drug. I have better examples of merged drug. Here I will show you, yeah. So just a little bit more about fragment-based and scaffold hopping. So this drug is a PI3K inhibitor plus mTORC inhibitor. I could not find a very nice picture of this one which will differentiate the two, but I made this picture. This is obviously incorrect because hydrogen cannot form a double bond like this. It is a nitrogen there, okay? But this is showing you a hybrid drug. One part is a cancer kinase inhibitor, one part is a HDAC inhibitor attached with a linker. This is obviously wrong drug. I mean, the molecule is itself wrong. But many compounds are dual PI3K-mTOR inhibitors or HDAC kinase inhibitors, just like a cartoon like there. True merged pharmaco, not a combo. So look at this. These are very good examples. So this is one part of the drug. This is paracetamol. Paracetamol is for what? Anti-inflammatory, antipyretic, right? So you mix them together, okay, and now you see the whole of it. So this is fusion or merged, or what kind of drug is that? The first one. Fused, because you are not really removing any part. So this is fused. These two together, this is really beautiful example of merged, right? Because this ring is actually becoming this ring. That's why they have changed it green. That's what I was looking for it yesterday. This is really beautiful. This is a perfect example of a merged drug. Works both ways. And this guy here, these three are together to make this, of course, merged, right? So you can see the examples of these drugs here, right? And molecular docking is very helpful in that case. So when you're developing those drugs, how many of you have used AlphaFold? The AlphaFold is different, but have you used AlphaFold? AlphaFold tells you about the protein structure, and you can take that structure and also do docking on top of it. But many of you do docking, right? So yes, so docking is very, very useful, okay? Now let's go to multiple target biologics. Something is missing here. This is number 5. Number 4 is missing here. I will correct it once I get back there. So again, you can use biologics to target many things. It's relatively simple here. You can make a bispecific antibody. One is targeting the cancer cell, one is targeting the immune cell. Bring it close. It will kill it. Here, this is a tri-specific antibody. It binds 2 different antigens and a tumor cell here. Okay, and this part will bring NK cells, right? I wanted to include this one to show you something very interesting. So I told you up to the nanobodies, right? So antibodies are 150 kilodalton. Then there are dye bodies. This course doesn't have enough time to go over everything, but then dye body, this is smaller. Then FAB 55 kilodalton. Then SCFV. Have you heard of SCFV? Anyone? I hope there is one person who has heard of SCF. Where? Where did you hear about it? CAR-T, not tandem. All CAR-T have SCFV. Please remember that all CAR-Ts have SCFV. The thing which recognizes the antigen, it's not the antibody, it is SCFV. Okay, so it's much smaller. How small compared to antibody? So this is CAR-T. Then we never talked about any of this except nanobody. So anti-calin, nanobody. Look at this thing. Nortin is just a tiny bit. There are 3 sulfide disulfide bonds which stabilize the structure. This is very stable. Just 4 kilodaltons, this thing. Okay, so we are coming back to fusion proteins. So remember I told you, you can have bispecific, trispecific, and fusion proteins. So these are fusion proteins. So monospecific, right? One binds, this is the protein of interest. This is your one compound which will bind the same. This is multispecific, binding 2 different proteins. If it is multispecific, see receptors on the same surface. This guy can bind receptors on the same surface, or receptors on different cells, or different binding sites. Like this part is binding here, this part is binding here. It can bind the ligand. So you can have all different kinds of combination. Okay. Now let's do PROTAC-based polypharmacology. So what are PROTACs? Remember about PROTACs, these are very important, right? What advantage PROTACs give to you as compared to small molecules? Yeah. Anyone? Why would you use PROTAC and not a small molecule? Go ahead. You can target the undruggable thing. Yeah, because it is hard to make PROTAC for everything, right? Sorry, small molecule for what else? You can make a small molecule with dual target too. Degrade the protein. See, small molecule, imagine I am the whole target here, right? So if you are inhibiting the thing, you are just putting something in the mouth like big rasgulla, which cannot be eaten, right? So you cannot hear the voice, but the whole thing is there, hand gestures are going on. But if the protein is doing a lot more than just the enzymatic activity, and it is doing a lot of interaction, making it disappear is very useful, right? So you degrade the whole protein. So that's why. So you can also do PROTAC-based polypharmacology. You can make one PROTAC which can degrade many targets. Okay. So for example, you can make dual targeted PROTAC which will inhibit CDKs and an oncogene. So look at this one. This one here, this target will inhibit 2 different targets, synuclein and tau. Okay, look at this PROTAC here. So this PROTAC is inhibiting one protein of interest and also the second protein of interest. So basically, the molecule which is there, which is binding more than one protein, and no matter which protein it binds, it will degrade it. So it is dual specific, or you can make 2 different PROTACs for 2 different proteins, right? They're attached to the same E3 ligand. Is it making sense to you? Keep in mind, you guys have your exam tomorrow, so pay attention to the details. Okay. When I'm asking you questions, that means these are my favorite questions. Okay. When I teach a class there, I always tell them, because no matter, even if you're teaching a semester long course, you tell a lot of things. Some of the things are not important from exam point of view. In my time, professors would never tell us what is important for the exam or not, and we could never dare to ask. Anything which is discussed in the class could be in the exam. But nowadays students want to know, and I tend to also say that I'm including some slides just for your knowledge. It's not going to be in the exam, right? Because when you show slides, sometimes it has more information than what you want to give it to the students. So I always tell them that if I say something is important, that means it's going to be in the exam. That holds true here too. So if I'm asking you about certain things, pay attention to those things. Okay, so you can see that, right? You can build protags which can attack a lot of family members altogether. So this is one compound. See, this is really good. This is the part which will inhibit two kinases at once, CDK5 and GSK3 beta. Okay, so this is a dual protag, and this is the one which will bring E3 ligase. Okay, look at this thing. It's just really good compound. Okay, at 42 nanomolar, it will inhibit GSK3 beta and CDK5. Then you can also do polypharmacology by hitting something. There is some kind of toxicity issues also, but suppose you hit heat shock proteins. What are the functions of heat shock proteins? Do you guys know? Have you guys heard of heat shock proteins? Tell me, Shivani. Conformation. So what heat shock proteins do, uh, huh? These are chaperone proteins, you're right, and they help in protein folding, especially under heat shock conditions. And also they help in the protein folding of many different proteins. So sometimes when you inhibit a heat shock protein, they have many client proteins. That means one heat shock protein may take care of 20 different proteins folding. And if these 20 proteins are all bad proteins, so most of them are bad proteins, you hit one heat shock protein and all the other proteins will not fold properly. If they are not folded properly, they will all be cleaned by the proteasome system. When the proteins are not folded properly, they are degraded, correct? Because the cells will recognize it. So this is a extremely dirty polypharmacology, very, very dirty, because some good things will also not get folded, right? It will have very high off target effect. You are correct. So this is like not the method of choice, but this is just to show you. Just whenever you see so many of them, it is scary. Okay, but this is also one way that you are just shutting down the whole pathway just by hitting one protein. Okay, now this is extremely useful, this one.
Shock proteins are those when you were discussing about transcription and plasmid incorporation. Is it
So that's you give heat shocks for 30 seconds to 45 seconds. So that is not enough to give a heat shock. I mean, they will take the plasmid in, but because what you do is that you keep the cells these are not rapidly going cells. They are frozen cells. So you put them in ice for 30 minutes. So nothing is going on. They are not dividing at 30 minutes on ice. Then you give heat shock, so they just suddenly take the DNA in because they were on ice for 30 minutes. Then you are putting at 42 degrees. Then you put them back on ice for 30 minutes. No, 2 minutes. And then you grow them. So by that time, the heat shock thing is gone. But what I was telling you about heat shock is that if you take E. coli, which is growing at 37 degrees, and then you put them at 42 degrees, then they will overexpress heat shock proteins to prevent the proteins from getting denatured. But these heat shock proteins are naturally expressing in the cells to aid in protein folding. These are not necessarily induced by heat shock in humans. These are human proteins, right?
the proteins which are not properly folded?
No, they are not degrading. It's the cellular damage control machinery is doing that. So when a lot of complex proteins are produced, heat shock proteins help in protein folding. Okay. Yeah.
If you want like gist of like what is the gist of the role like heat shock protein? If you want to know it in like few lines.
So few lines it is this that heat shock proteins are chaperone proteins which help in protein folding. Okay, that's the main thing. In E. coli, it has a different function altogether because some heat shock proteins are produced when you put E. coli growing cells in under non-physiological higher temperature. So then these proteins are produced to help to prevent other proteins from getting denatured. Make sense? Yes. No. Heat shock proteins are produced across the board from E. coli to humans. What happens is that, so here is something, I'll tell you something very interesting. So as RNA is making, whereas ribosome is making the protein from RNA, right? So as the protein peptide is coming out of the RNA, it slowly gets folded, okay? And the whole protein is folded and final protein is made. If there is some mutation in the RNA itself, then the protein will not fold properly because the protein will also have the mutation, correct? So then, and if it doesn't fold properly, it will aggregate, and the cellular machinery will clean up the aggregate machinery. It's like um, it's like trash. So trash is cleaned, right? So there should be no aggregation. So that is the way, if RNA is messed up, the protein will be messed up and they are completely cleaned. So we don't have any damaging effect. But if the DNA is messed up, it's always there. So then it is a problem. That's why people say genetics is messed up, that can give rise to disease. RNA messing up doesn't give rise to a disease unless it is a microRNA. Okay. So heat shock proteins are needed for some proteins to fold them properly. So as they're coming out, they still need assistance from heat shock proteins to fold properly. So in cancer, it is shown that many oncogenes, these oncogenes are not natural proteins, right? They have deleted some part, they become hyperactive, they have undergone mutations. So they need assistance from heat shock proteins in properly folding. So the idea is that if you inhibit heat shock protein, you are inhibiting many proteins at once. The stability of many proteins at once, and if they are not folded properly, they will all be cleaned up. Is it making sense? Okay. I will tell you something very interesting here too. Do you guys know about codons? Have you studied genetic code? All of you, every single one of you. Have you guys heard of codon optimization? No. Okay, so can you name any one amino acid which has 6 codons in the genetic code? So methionine has one codon, AUG only, or ATG, whatever, right? It has only one codon. Tryptophan has only one codon. Glycine has 4 codons. Alanine has 4 codons. Leucine has 6 codons. Arginine has 6 codons. Okay? Why I know all these things? Because I teach this thing. Okay? So I will tell you a very simple example, and that will tell you something very interesting. It was a science paper. Okay? So glycine has 4 codons: GGA, GGC, GGU, and GGG. Okay, 4 codons. So let's say in humans, not all 4 codons are used. Let's say, for example, for humans, GGG is the predominant one. 90% proteins, if they have to make glycine, they will use GGG and rarely GGA. Let's say, this is hypothetical part. Codons are correct, but which one is used more, that I don't remember. Okay, but in E. coli, GGA will be used. Okay, so if I'm making a human protein in E. coli, okay, so human gene has GGG because humans like to use GGG for making glycine, but E. coli likes to use GGA, correct? So what people do, you pay a company and you say that you codon optimize it for E. coli. So it's a human protein. So human protein will have GGG, for example. So it will say change it to human can also use GGA. Remember, it's a universal code. We have the same code as E. coli. But because E. coli have more tRNAs for GGA than GGG, they will say, okay, optimize it so that E. coli can make the human protein efficiently. Once the protein is made, GGA, GGG doesn't matter, right? Because it will be glycine. Do you guys understand what I'm trying to say? You guys are able to understand? Should I repeat it? No, you got it, right? All of you. So some scientists were doing very simple experiment thinking of publishing the paper in two impact journal, right? They were just trying to make a protein. They just give the company some money to codon optimize it. The company optimized it and then they express it in E. coli, and the protein never formed. Never formed. It would just form aggregate and just crash out, crash out, crash out, crash out. So they could not understand. So then they went back to human cells and then they optimized it perfectly for human also. So what they did is that wherever they had GGA for human, they made it GGG, thinking that the protein will be expressed better because humans like to have GGG, but the DNA sequence had GGA at some places. So they changed it to GGG. It would not express in humans also. It was just degraded. The moment it was expressed, it was degraded. Can you explain to me why? So let me summarize the problem. So there is a DNA sequence, and let's say there are 10 glycines. And humans, we know from before that humans like to have GGG for glycine. But in 10 places, 8 places had GGA, which is not liked by humans to express glycine. So these guys made all GGG, all 10 of them, and the protein would not express. And we know that humans have tRNAs for GGG more than GGA.
Does it need C because it is triple bonded with C? Does it have something to do with stability?
Codon has nothing to do with triple bond because it's not double stranded. It is just single stranded. So there is no hydrogen bonding that way. So it's not that. So think bigger because, like I said, this paper would have come in a two impact factor journal. It came out in Science because it was such a basic thing which was going wrong there. So I want you to think big picture. Think it like, think of it as if you are witnessing the whole drama in front of your eyes. So let me repeat it once again because I want all of you to think about it. So because you guys are scientists, you are in research, you have to, if something so strange comes out, so suppose you had all GGG wherever glycine was there. tRNAs are the one which bring the amino acids. So if you have better tRNAs for some codon, it will be faster synthesis because you have supply, ready supply, right? So the parental DNA which you isolate from the organism at 8 places, let's say they had GGA, which is a bad codon for glycine for humans. And they were making it in human cells. So they gave it to the company to change it to GGG, which is considered a good codon for human. But the protein would not express. It would express from the bad codon. Like if they use the original DNA, the protein would be made, no problem. But when they were using this codon optimized one, which is supposed to be better, the protein would not express. Same cell line.
Because it's coming from the DNA.
No mutations. We have introduced mutation, but these are functionally silent mutation, right? Glycine is encoding for glycine. So you cannot even say that the protein, you have introduced a new amino acid and protein is not folding properly. You cannot say that because glycine remains glycine. You are just changing the codon. No, it is a mature, when you do the plasmid, introns, exons don't come into picture. It's just the DNA directly making the RNA.
Is it just because coming from the mother
Why that is making a difference?
Is it their identification that if this proportion will not?
No, what identification? No, so think about it a little. I want you, like when you are in the jungle, right? You are looking at one or two tree, but if you go up, you can see many trees, and you go up like a bird's view, you can see the whole jungle. So I want you to come out of that thing and see what's going on. Tell me. No, so GGA and GGG, they are both glycine. So suppose I make from GGA, I make GCA, then I'm changing glycine to alanine. Then of course it will change the protein folding because you are putting a new amino acid there. But the thing is that they have not even changed the amino acid, they have just changed the DNA sequence.
Ma'am, if I'm not wrong, the problem is we have a continuous strand of GGG for the 10.
No, it is not continuous strand. It is a long DNA, and let's say it has 10 glycine. I don't know what was the exact issue in that protein, but let's say it has 10 glycine. So at some places they had GGC, which is not such a uh, good codon for humans, and the answer is hidden in this sentence, which is not such a good codon for humans. They replace it with GGG, which is a very good codon for humans. So tell me, I'm going to guide you to the answer. Go ahead. Can you, if you speak the right answer, I want to have you this thing.
Maybe it will interact with some other amino acids of the DNA. Let's suppose if it is like at 50th position, so it's not like it will interact with only the nearest one. Maybe when protein fold, it may interact with some other, like maybe 70, 80.
No, you are right in that respect, but the thing is that glycine will still remain glycine. So 50th position was GGC, right? And we changed it to GGG, but it is still glycine. When
Yes,
it
ma'am.
was GGC, it was glycine. When it is GGG, it is glycine. So if it was interacting with the 70th position, it will still interact with the 70th position. You are going in the right track. You have to just go a little bit
Yes.
further.
I think it is coding for glycine only
Yeah.
right now also, but I think it's the interaction, like the interaction which are going on inside that. Like maybe A will interact somehow differently with other amino acids which are present, and it will determine the aggregation of that. Like it may be degraded, like the protein, like the
So
place where
if
G
the
was supposed to be, there is A now.
no, that's what I'm saying. You are not changing the protein sequence at all. You are not changing, it is identical. Okay, so I will give you a clue because I won't give you the answer. I will keep on giving you the clue till you guys go. Just give me, yeah, go ahead. No, they don't form dimers.
Does it have something to do with GGA when it's about folding?
GGA will always bring glycine and GGG will also bring glycine. You guys are coming very close. You are almost at the edge of the door, but you are not getting the whole picture. So, okay, now I'll give you a clue and all of you will get it. So what happens if you use a poor codon? What will happen? It will still encode for glycine, no doubt about it, right? Because it is glycine. But what will happen? No. As protein synthesis is going on, you guys know how things work, right? So tRNA has to be there. But if there are not enough tRNAs for GGC and there are too many tRNAs for GGG, so anytime GGG comes, protein synthesis will be faster. Anytime GGC will come, protein synthesis will be. So nature has made it slower at that point because that was an important junction where it needed to slow down for proper protein folding. Okay, sequence is the same. So it had to slow down. When we were putting GGG, it was just so fast. The whole thing was aggregating. It was getting cleaned up. Protein was degraded. So nature has chosen for it to slow down there by using the worst codon. And when these guys did codon optimization, that's how it became a science paper. It was such a new discovery, and it was a recent science paper, like 4 or 5 years ago. So just imagine slowing down also at places. Suppose you're driving a car and you're going over the hill where there's a real danger, you have to slow down. So proteins folding at that juncture was so important, it needed to slow down. Okay, so my point here is that if you see something so unexpected, right, when you're doing the good thing and something bad is happening, some nature is telling you something very important. So look at the big picture, step out of the problem. Somebody I met and we were having dinner together, she's the director general of something something of cybersecurity, and she lives in Bangalore, which is a very nice city, and she was posted in Delhi for 4 years. So the first question I asked, so how is the pollution? And she paused for a minute and she said, when you are within the problem, you don't see the problem, right? So when you're too deep in that problem, you are not thinking about protein folding. So you need to step out of the problem and see what is going on. Let me see. Okay, so that is there. And now you are watching, observer, right? Have you heard of the witness thing? You witness the thing and don't become part of the story. That's the witness thing, right? As a scientist, you just witness what is going on there. Okay, so now you guys know what it is. We had a fun discussion, right? Okay, let's go to the main story here. This is very interesting. Phenotype driven polypharmacology. How many of you have heard of forward chemical genetics versus reverse chemical genetics? Nobody has heard of it. This was a very popular term in the late 1990s. So what happens is that there are two ways to drug discovery. You say, oh, BCR-ABL is there, and now I'm going to make BCR-ABL protein pure, and I'm going to screen millions of compounds against this pure protein. So the goal is to inhibit BCR-ABL. So that is called reverse chemical genetics. You start with a target, and then you screen millions of compounds. Forward chemical genetics is that I don't know the target, but I want to kill the cancer cells. So I take cancer cells and I screen millions of compounds. And then I choose those compounds which kill the cancer cells. And then I take those compounds and I use it against normal cells. And I want normal cells not to be killed. So then some will kill both normal and cancer cells, we throw them out. But those compounds will just kill BCR-ABL cancer cells or any cancer cell and not normal cells, they are my hit. That is called forward chemical genetics. Okay, forward chemical genetics is phenotype. Okay, I want phenotypic change, live, dead. Like this kind of cell versus this kind of cell, that is change in phenotype. Phenotype is how we look, right? Our DNAs are 99.99999% same, right? Anywhere in the world, if you compare to any human being, right from somebody in Antarctica, our DNA to the DNA of those people, 99.9% identical. Phenotype, entirely different, even in this small room, right? So DNA is same, phenotype is different, right? So when you are doing phenotypic screen, you select drugs by outcome, not known targets. So then you can understand polypharmacology. I want to kill cancer cells. If it is hitting 10 targets, 20 targets, I don't care, as long as I'm killing cancer cells. So that is the most important thing, phenotypic driven polypharmacology. Okay, I'll come to that in just a few minutes again. Like she said, Janvi, drug repurposing. That is, so you, this, excuse me, target this for one disease, some drug was identified. Okay, but we also said, okay, in this disease, it is hitting this target, but this is the side effect. But if the side effect target C is cause for another disease, you can use that for that disease. So look at this thing, how long it takes for the drug clinical trials. You do basic study, you do drug design, you do in vitro preclinical studies, then phase 1, phase 2, phase 3, and then the whole thing. But if you are taking a FDA approved drug and repurposing, it takes really a short time. Sometimes, in COVID time, the pressure was so high, people never thought of mRNA vaccine, but people were dying across everywhere in the world. It was just not ending. It was just unbelievable everywhere. Like, people are very nice and very considerate under better circumstances, but when things go a little bit haywire, like everybody was breaking every rule, right? So we were under tremendous pressure to find a drug, and so we made the discovery we would have never made it at that time. Like, there was so much pressure, right? So at that time also, AI helped in drug repurposing, taking all the FDA approved drug. Like, every lab was working on COVID at that time, just trying to do their part. Okay, so success story, of course, metformin is one of the best story. There was this thalidomide thing which were given to pregnant women, right? Now it is given to multiple myeloma patients. Okay, then there is another thing called chemogenomics in polypharmacology. So here what you do, you take a whole compound library, one compound at a time, and then you add it to the cells and you do the whole pathway analysis like genomics, proteomics, metabolomics, RNA seq, everything to see exactly what are the different things it is hitting. Like, you get the whole profile of it, and then you can plan which drug can be used for drugs. But the thing is that this gives you so much information that it is hard to like make sense of this much in this thing. So that's where artificial intelligence really comes into picture. Okay, that is what we will see in deep medicine. So this is like a whole library of compounds. You can give genetic treatments, you can give small molecule treatments, and then you can look at many different, and you can build up network, and then you can do that. And then finally we'll come to AI and data-driven approaches. So our 30th lecture is all about AI and data-driven approaches. So when we started the lecture, we were talking about conventional therapies for cancer, like chemotherapy, right? Chemotherapy, surgery. Then we went to precision medicine. And AI is beyond precision medicine. AI is that, will not let the disease develop. Precision medicine will only come once the disease develops. So AI is supposed to be proactive and telling you years in advance that there is this risk factor and the disease never develops. Okay, so that will come. So AI and data-driven approaches. So drug target interaction prediction, it can do multi-target design and optimization. It can repurpose existing drugs, predicting adverse effect. So many things at one it can do. Okay, and then you can find exactly the right compounds. And AI can even dream up molecules and even do the synthesis of molecules. Have you seen or have you heard of labs where people are doing synthesis all by robotics? Anyone in IIT Indore? Yeah, but I went in 2016 or 2019. I think in 2016 I went to IISER Bhopal and IISER Pune and IISER. I went to many places. I went to 10, 11 places. I don't know which IISERs or which IITs were there. I think these were IISERs. IISERs are newer than IITs.
There is one professor in CSR IICT Hyderabad. He is working on like during one workshop he showed his model.
Yeah, so it is, but I have not been to Hyderabad in any place in Hyderabad, but I went to some of the ISR, and in that lab, everything was done robotically. The compounds was purified by the machine, synthesis was carried out, everything. There was just barely any human interaction. There is a big company where I live, 1 hour away from that. You might have heard of Eli Lilly. It's a world famous company. So they are saying we don't hire synthetic organic chemists. It's all done by robots. There was a time when synthetic organic. I was a synthetic organic chemist by training from PhD, and they don't hire them because it's all robotic now. So we used to spend hours and hours doing column. This person at ISL said, who does column now? The machine does it for you. And those who are organic chemists by training, oh my God, it's a lot of sweat purifying by column. It still gives me nightmare. Like you are so patiently doing it, and you increase the polarity. Impurities come together. If you have done organic synthesis, you know that it is so painful, right? So, but basically, this is where the science is going. Okay. So for AI to work, you have to feed in data. If you don't feed in data, so that is why that all of these things add up to that one. And all the omics data, genomics, epigenomics, transcriptomics, proteomics, metabolomics, they all have to add. Remember, anytime it is omic means global, across the board, right? And all of this thing for each of us has to be fed in the AI, okay? And then they can actually give you the exact drug for that person. Okay, the more targets you hit, the higher the chance of side effect. So using AI to explicitly filter out anti-targets. Now I'm going to show you some case studies. So imatinib and dasatinib. Dasatinib is just a next generation imatinib. So when imatinib is Gleevec. When Gleevec was given, it was a huge hit, but then patients developed resistance very quickly. So then dasatinib came into picture. So this is the parental compound. This is the next generation compound. So this was designed to target BCR-ABL, c-KIT, and PDGFR. This accidentally polypharmacology allows it to treat gastrointestinal stoma. So this was accidental. It was people did not know about polypharmacology at that time. This was just a really good luck there. But the dasatinib was deliberately made it dirtier, so it inhibit BCR-ABL, SARC, c-KIT, and ephrin receptors. So when people develop resistance towards Gleevec, this drug was much better. Okay, this is a real thing. So remember we were saying PD-L1 inhibitor or angiogenesis inhibitor. This one, sunitinib, this inhibits so many targets at once. Remember, angiogenesis inhibitor receptors are VEGF receptors. So it hits many VEGF receptors, and these are the ones which cause tumor growth, cell proliferation. So sunitinib hits so many targets at once. So it is inhibiting tumor growth plus angiogenesis. It's not a PD-1, PD-L1, but it is inhibiting tumor growth and angiogenesis. So this is a very, very powerful drug, sunitinib. Then lenvatinib. Lenvatinib, look at that. So many of them. It is increasing. Okay. So it is inhibiting cancer, cell growth, as well as angiogenesis. So look at that, so many of them at once. Then sorafenib, it inhibits RAF kinases, VEGF, BTGFR. It's really good. Anytime you hear VEGF, that means angiogenesis. VEGF receptor is the receptor for angiogenesis. Okay. So these are some examples. So these drugs and how many targets they are hitting. So they are hitting several of them, and they are extremely effective drugs. One pill, so many things. Okay. Then I was telling you about schizophrenia, right? So there is this drug clozapine. Look at that. How many of them? It's unimaginable, and that is really a game changer for this disease. Brain is really complex, right? This morning I was working on a lecture 30th. I had prepared it yesterday, but I keep on changing it till the last minute every single time. So I was adding something, and I was talking to my niece, and she's in class 12th, 11th, 12th. She's going to apply for college, and she has to write an essay, and she was telling me that she wants to be a doctor in neuroscience, and she said she wants to write an essay on neuroscience and AI. And she told me some really neat ideas. I have never thought of those ideas. And she brought AI, and I was working on AI as like I was putting things on the AI. So I said, I will send you my presentation, but it is cancer in AI. So you read about it. But she gave me some really nice ideas about her own thing, spatial, like, the brain is three-dimensional, right? So, how memories are stored, how do we see space? How do we see the depth versus this? And how we are moving hand in three dimensions. And when somebody has prosthetic arm you think about it, and your arm is moving based on your thinking. So those kind of things she was telling me, and how you can use AI to do that. So it's just unimaginable the kind of things you can do with AI now. Okay, so obviously, when the drug is clean, it is much easier to get FDA approval. But now FDA is understanding that dirtier drugs can be, agencies are adapting. Okay, if a dirty drug saves lives in a complex cancer where clean drugs fails, it gets approved. Okay, so AI designed multi-specific. Okay, so we will move from discovering accidental polypharmacology to engineering it. So Gleevec was accidental. Okay, I have nothing to do with Gleevec but I just joined when Gleevec was getting approved, and I was part of the kinase group, so I got all the perks of the approval of Gleevec without contributing towards Gleevec. Like, there were just huge celebrations for Gleevec. And these are not parties, these were taking people to Mallorca in Spain and taking a trip on Mediterranean Sea where they were serving you finger foods all day. And while your ship was going over the Mediterranean, and Mallorca is the most beautiful part of Spain, it's on the Mediterranean coast. So I enjoyed all the things. I did not contribute at all towards Gleevec. Okay, but I was just there at the right time to enjoy. Yes, yes, yes. Okay, so, and we can make obviously Protax. Molecular glues are something similar to Protax. Protax, in very simple words, Protax have a linker between. But if you remove the linker and those two things are together, that's called molecular glue. That's the difference. So now this is becoming very popular. Digital twins and avatars. What are digital twins? So suppose I am there, right? So they do my proteomics, genomics, metabolomics, transcriptomics, everything, all my history, everything is there. So they know me. Then they create a virtual me with the exact same chemistry, PGX, my genetics. They know what drugs will. And then they will test the drug virtually to see what kind of interaction it will have. And then they will give it so that they can predict everything in advance. Okay, so this is personalized. This is not precision. This is personalized. N is equal to 1. 1%, 100% personalized. 3D poly plus dynamic modulation. So this polypharmacology is not lack of precision, it is precision applied to the reality of biological complexity. I hope you understand the meaning of this thing. Precision means you're precisely targeting one. But polypharmacology, you can say it is not precision because it is going to many targets. No, it is extremely precise in choosing all the right targets at one, because biological systems are very complex. You cannot survive with just one target there. If you have to change a nation, you have to, do it in many different ways. Okay, so that's it. What should we do? Our lunch is at 1 o'clock. It's 12. So it's very far away. Okay, let's do the next lecture then. Okay, do you guys want 5 minutes break or should we go? Break? Okay, 5 minutes break. So it's 11:46. So we come at 11:51. Okay. Oh, that's why today we had 3 lectures instead of 4. That's why it is going to go fast, right? Okay. आपने बंद कर दिया ना? हाँ ठीक है. So today we can even finish at 3:30 because all of you got the lecture notes for no, Professor Venkatesh said it while when he was sitting here. I saw it in my watch. Actually, thought I sent to him on what day is it? It was Wednesday. I sent it to him at 4 o'clock on Wednesday, but actually it got stuck in my draft. It never got sent, and I was waiting why Venkatesh is not sending it to you guys. Then I realized it's my fault. So I sent to him this morning because you have to have that for your exam tomorrow. Yeah. See you later. See you later. See you later. Yeah. बंद है ना अभी? हाँ. Today the driver was asking me what should I come, when should I come, and I said 4:30. I should have said 3:30 because it will be done by that time. I'll tell Venkatesh to call him. Seems like such little work, like 3 lectures seems, oh, it will be done soon. That's the end of the story. क्या फटाफट चला गया, पता भी नहीं चला. मेरे पास एयर इंडिया का रोज आ रहा है अब जो है ना, मतलब अभी भी आए हैं, अभी भी वो योर अपकमिंग ट्रिप, डू दिस डू दैट, जस्ट रिमाइंडिंग मी टाइम टू गो. Actually, I wanted to leave on Sunday because this thing is done on Saturday, but there were no tickets available on Sunday as well as on Monday. क्योंकि ना ये Christmas trust between patient and doctors. What does it mean actually? It means is that AI is not going to take our job. Okay, we have to collaborate with AI. AI is our assistant or collaborator in giving the exact right thing to the patient, preferably to a healthy person, so that the disease never develops. That's the thing. Okay, so we started with really bad therapy, then we came to precision therapy, and now we don't even want precision therapy, we want predictive therapy so that the disease never develops. Okay, that's where AI can be very useful. And now we don't want precision therapy, we want personalized therapy. Only for me, only for you. So your genetic makeup will determine exactly. Okay, and it can tell you in advance, years and years and years in advance about a possible disease. Okay, so evolution of therapy. Empirical medicine is that you already have a symptom, and based on the symptom, that is, they are giving you the medicine. Precision medicine is looking at single biomarker, HER2, this thing. What is deep medicine? Deep medicine integrates multi-omic patient data plus AI to deliver, this is the beauty, predictive, preventive, personalized and adaptive therapy. Explain to me, what are these three? Predictive, preventive, personalized and adaptive therapy. This is a really beautiful sentence. Tell me, what is personalized? According to one person, not a group of person, one person. What is precision therapy? Subgroup of people. We want exactly one for me, one for you, like that kind of personal. N is equal to 1. What is predictive? This is the keyword, before the disease happens. That's the keyword, predictive, before the disease happens. Preventive, we are not going to let the disease come to you. See how beautiful it is. We don't want to be treated. We don't want the disease. Correct? Predictive, preventive, personalized, and adaptive. What is adaptive therapy? Say that again.
No, the drug is completely accepting that patient. Its metabolism gets adapted to what the drug is.
So all of you are correct, but the best definition is adaptive, what Venkatesh said, or you said. it is not metabolism, it is that as the disease changes, your regimen is also changing. So it is continuously monitored and changing according. And so if the resistance comes in, before the resistance comes in, you change it. You don't even let the resistance come in. So that is adaptive. It's not you give PD-L1 therapy and forget about the patient. You are just constantly monitoring and changing it. Okay, so for that you really need multidimensional data, correct? That is deep medicine. So precision therapy and artificial intelligence, you do precision therapy plus artificial intelligence is equal to deep medicine. Okay, so there are many ways to do that. Deep phenotyping, we are going to go through all that. AI-enabled target discovery, matching, adaptive therapy, polypharmacology, predict, personalize, prevent, and adapt. These are the 4 pillars. Okay, we don't want precision medicine. We don't want disease to start with. So there are 5 things: personalized, one individual; precise, exactly where to tool; predictive, preventive, participatory. Okay, you are empowering patients to make the decisions. You are just giving. So here, for this to work, for AI to work, you have to give as much data as possible, correct? So you are getting clinical features. When we get pathology done, right? So if they are doing imaging of our skin, so that is called immunohistochemistry. Histo is tissue, immuno is you are using antibody and you're looking at the whole thing. So it is immunohistochemistry. So when we as humans see, we can just, our brain is deciphering only this much information, but AI can see 10,000 times more information in that. We are not able to see that much or decipher that much, right? So pathology reports the raw data. What AI can see, human brains cannot even comprehend. It can look for so many details in one report. So for pathology or radiology images, they can do wonders there. So that it says digital pathology, radiomics, genomics, epigenomics, transcriptomics, proteomics. The more data you give, the more intense they can study you. They can read you completely. Genomics, they can tell you PGX, which drug will work, which will have side effect. Okay, so it is completely precise. So defining deep medicine is the convergence of artificial intelligence and precision medicine. If it represents the ability to digitize the human being in high definition, like they have all the information about you, each and every piece of information. The people get very afraid with AI. The goal is not to replace doctors, but to provide with them with the gift of time and superhuman sight. We will talk about the positive and the negative things of AI here. Okay, so here is the problem. So we go to the doctor scribbles a note, right? It scribbles a note. But the thing is that your health is with here, his health is there, his health is there, his health is there, my health is there. They are not integrated in one place. And even if it is integrated in one place, it's all written in different languages. I'm not saying Hindi versus English, but it is scribbling here. So for a computer to read it, it has to be the computer language where the computer can make sense of it. It has nothing to do with Hindi, English, or something like that. So there is a huge data tsunami. Just imagine the amount of medical data doubles every 73 days. Doubles worldwide every 73 days. But the thing is that it is not organized, it's not in one place. And then there is always a diversity issue, health disparity. Like, people who get checked, there's huge amount of data. In Europe, I hear the healthcare system is outstanding. So there they probably have a lot more data than other countries. Okay, in US at least they do all annual checkup free. So most of the people get annual checkup. But the data are all in different places, right? So, and 80% data is unstructured data, notes, image, genomics. People have it in their own possession. It's not part of any system. It is unstructured and unusable by traditional thing, and people make one mistake in every 4 cases. That's a huge 25% error. Sometimes it is due to workload, sometimes they just don't have the ability to analyze the data properly. Okay, so the limits of human cognition. So human physicians are excellent at pattern recognition in low dimension. Oh, you have red rash or fever, together you have measles. Okay, but when it comes to, tremendous amount of genomics data, proteomics data, people don't have understanding of it. Okay, and they cannot diagnose the data. See, here is the thing: when we didn't have genomics, proteomics, metabolomics, transcriptomics, okay, they will look at your rash, that's it. Or they will look at the fever, or they will look at your throat. Okay, that time that was the best, but now it's different. Now we have huge amount of data. It is a piece of cake to generate this data for any human being. You take just the mouse swab, you can do the genomics thing, right? So now it is possible to get the data, but we don't have the ability to interpret that much data and make sense out of that data. So that's where artificial intelligence comes into play. Okay? So that is where biological complexity exceeds human capacity. Okay, so look at that. So many different inputs are there. I briefly told you about case studies. That's people who live in Chicago, close to highways and everything. They have high blood pressure, right? So there are environmental factors, there are social factors, there are stress factors, there are financial factors. Then there is obviously genetics, right? Remember I told you about exposome, environmental thing, how much it affects, right? So all of this, family history, cognition, state of mind. So there are so many things, and that makes a person. Okay, I have seen in my own case that I am very allergic to some very commonly taken drugs. Like I have very strong reaction, and doctor has actually told me I've never seen this thing, but it happens with me. Okay, like there is a headache medicine, she gave it to me and I took it first time. So I was feeling like really bad, like extremely bad. So then I did not take it. Then I had a bad headache and I took it. I said maybe there was some side effect. I took it again. And I was living in an apartment, so it was late at night and my condition was so bad. I opened the door of the apartment. Went out in the open area, like I thought I could not breathe. After that, I never took it. And it's a very, very famous migraine medicine. Everyone takes it. So I obviously have a different PGX and I cannot metabolize that drug. And I've seen this for many things, and I'm sure all of us have our unique things. So you cannot give really bad cancer drugs without knowing the whole history of that person. Okay, so this is not a trivial thing. I'm talking about a headache medicine here, but imagine for serious diseases, right? So the solution is artificial intelligence. It can synthesize huge amount of data. It can tell you the treatment, it can tell you the side effect, all of these things. However, for AI to work extremely efficiently, you have to feed in more data to that. Like it has to have a lot of things. It has to have the results. Okay, this person with this genetic makeup was given this drug and this is what happened. That's how it is built up. Okay, so you have to do. So do you know the difference between machine learning and deep learning? Machine learning is that you tell the machine, look for this area. So there is a human input there. But deep learning is no human input. You just feed the data, it knows what to look for. It will keep on re-educating itself as it goes along. Okay. So I will tell you about the core components of AI-driven polypharmacology. So we start with deep phenotyping. You will really love this part here. So what is deep phenotyping? As many data points as you can collect. So you can do all different kinds of things. We have discussed, you can look at the imaging, exposome, how the environment is affecting, cognitive health, blood analytes, whole genome sequences, microbiome, self-measurement, cardiac health, everything. All of the things. Okay, that is deep phenotyping. Okay, obviously it integrates all of these things. So patient is a data ecosystem. You're looking at the whole thing. You may have heard of these kind of stories that, identical twins, when they were separated at birth, One grew in one environment, the other grew in another environment. Same DNA, exactly the same, and completely different personality emerged. So it tells you about the environment, how much it is affecting, right? That depends on social interaction, the food, everything, right? And you can make two entirely distinct personalities out of that. And I know one of my colleagues, she has identical twins. They are living in the same home, same food, same parents, same love, same school, everything. One is more aggressive than the other, identical, completely identical. So the one who is less aggressive, the parents have to pay attention to, the otherwise the same guy will beat up the other guy, right? So you never know what could be the trigger there. So what is the fuel for deep phenotyping? Of course, the more data you feed in, the better it is. So all the omics things add up here. Now, target discovery. So all the things that we know AI can do. AI can do molecular docking, structure-based drug design. It can feed in all the things. It can identify the target. It can identify drug-target interaction, the toxicity, the PGX, everything. All the things that you studied, everything together can be fixed by AI. AI can even make up the whole molecules, dream of molecules. Okay, make up this kind of molecules, so the robotic systems can actually synthesize these molecules for you. Okay, in my time, synthetic organic chemists were paid huge amount of money because they were really in demand. Now they are becoming obsolete by this thing, right? I am hearing that AI is slowly taking over the IT industry also. And IT industry 20 years ago was like unending, not 20, 30 years ago, like Silicon Valley, you have heard how much. Now AI is taking over that thing too. So as humans, we are afraid of AI to some extent, which is okay, because AlphaFold, AlphaFold is a program to determine protein folding structure. So that's risk prediction and prognosis. This is the best part. Okay, this is electronic data. EHR is electronic something data. The full form is written electronic health record. Very good. It is electronic health record. So if you give all of this thing to that, it can do risk prediction and prognosis for you. Okay, this is the best part for that. Early detection and screening, like I said, mammography, people who have diabetes become blind over time if it is not treated. So all of these things can be predicted long in advance. I have met many people. There is an IT person in my department. So he just went for the annual checkup. He had no health issue. And he came to my office and he said, "Oh, I wasn't here for 1 month because I had surgery." I said, "What happened?" He said he just went for annual checkup. And remember, I tell you, they give money for annual checkup. If you do annual checkup, they actually pay you money. So people go for annual checkup because they get the money back. And in that, some cancer was diagnosed. And he got the surgery done, it was gone. So he had no symptom, but the blood test, you remember I showed you blood test can show a lot of cancer. So that cancer was predicted and basically it was prevented from really becoming aggressive. So early screening is very important. Okay. So diagnostic and imaging, this is where AI is outstanding. So I have seen hundreds of ISC slide, and I'm not a pathologist. Pathologists can see a lot of things, but AI can see thousands and thousands of more things there which we don't even know about. So they are extremely good in reading X-rays, CT scans, MRI, biopsy slides, all of these things, skin lesion analysis. Okay, so to enhance diagnostic accuracy and speed. So they extract hidden pictures, hidden features which humans don't even know. Like we don't even know what are the things which are hidden there, and they can read it and they can predict tumor genetics from a CT scan without cutting down a patient. Okay, digital pathology, they are extremely good. AI is extremely good. Okay, AI enable predictive treatment measurement. they can use the best, they can find the best drug or drug combination for us based on our whole virtual avatar. Okay. They can also predict the response in advance. Because we have this genetics makeup, this is how we are going to respond. Okay. So they can create a digital twin based on all our data. Just imagine if you give all your data to an AI, there is nothing hidden from the AI. They know about your environment, they knew everything about you. Okay. And they can create the virtual you and test it whether it will work or not. Okay, or what will be the side effect. Adaptive therapy, as I said, they can continuously adapt based on your response and keep on changing the therapy to the best of you, right? Like suppose you're responding, how to measure the dose or give this in advance or remove this because you don't need that. So it's a closed loop. As disease is getting, you are changing your therapy accordingly. So this is AI enabled adaptive therapy monitor, and then it is completely adapted to you, and then you can keep on adapting to you as you are getting better or hopefully not worse. Continuous monitoring is like, the smart watches we wear. It keeps track of our heartbeat and everything. Okay. People use glucose monitor and other things. Okay. What is your exercise pattern? Many times heart diseases can be caught in advance because they look at your heartbeat. Okay. A vocal biomarker AI analyzing what snippets to detect early signs of Parkinson, Alzheimer's, or depression, or microtremors. Okay. Now, AI can aid in clinical trial optimization. So clinical trial use AI to scan electronic health records and identify eligible patients for trials. Okay, so when you do clinical trials, right, you try to bring in a lot of diverse population. First of all, you can see what is the effect on diverse population. Plus, you also want to see what kind of resistance will emerge and how to connect it to the person's phenotype and genotype and all those things. So AI can really help in this kind of thing. Okay. And it can, of course, do predictive modeling. Drug repurposing, AI is very good in doing drug repurposing, just virtual screening. And it can say, okay, this drug might work for this disease. Okay. Now, pharmacogenomics plus AI. You know what is pharmacogenomics, right? What is pharmacogenomics, PGX? Guys, you actually have to study today. Many of you will get the, what is it called, certificate tomorrow, but my question paper is a real question paper. It's not a fake one. What is PGX? Pharmacogenomics. That is not the only thing. Yeah, predicting drug action based on the genomics. Like I said, I could not take that migraine medicine and rest of the world can take it. Something is different about me, right? You guys should also know about at least one case study for all of them so that you can give an example of something. Okay, pharmacogenomics is very important. Okay, so AI can of course predict pharmacogenomics. That's a piece of cake for AI. AI can design polypharmacology, which compounds, how to do it, right? So it can actually make the multi-target molecule to do that. Okay, so I wanted to give you, I have told you many things, right? What actually have been achieved so far and what remains to be achieved. Okay, so we already have AI working where we can do diagnostic much better. So we give the data, the pathology report. These pathology reports are not the ones you guys get or I get. Like we are the patient, we get it, okay, blood hemoglobin is 17%, this is that person. That's not the pathology. That is, AI sees the actual raw data. Okay, you get here, this is this person. That is useless for AI. It is like I am giving you the answer. I'm not showing you the raw data. If I show you the raw data, then you will make your own judgment. You will not depend on me. I'm not going to feed you that. So you have to show the raw image to AI. And that's when it can read a lot more than humans can read. So that's how the pathology, the diagnostic pattern is very good. It has enhanced imaging recognition. It can read a lot of things which we cannot read. Okay. Deep learning is widely used in radiology, X-ray thing, okay, pathology and ophthalmology for image interpretation, early detection of Alzheimer's and Parkinson years and years before that. So, for example, do you know what is the hallmark? How do you know a patient has Alzheimer's versus Parkinson's disease? No, don't tell me the signs. So if you see a Alzheimer's patient versus a Parkinson patient, how would you distinguish these two people? Exactly, hands like, voice will be slurred because they have Parkinson, correct? So they have motor defects and Alzheimer's have memory defect. So when a Alzheimer's patient has memory defect, right? So that is irreversible. You cannot reverse memory, this thing. Okay, it is irreversible. Once the neurons are gone, they are gone. By the way, I personally know of a person who has reversed Alzheimer's, which may be the only one in history, and she's a very close person. She's extremely close to me. Okay, so I don't know how she has reversed Alzheimer's. Okay, I will tell you guys about that story um, during lunchtime. Okay, but Alzheimer's is not reversible. You can slow it down, you cannot reverse it, you cannot cure it. So Alzheimer's, once the brains are lost, it is irreversible degeneration. So imagine, but Alzheimer's takes 10-15 years to develop. So before it develops, if you can diagnose it, before the memory is lost, right now we don't have any way to do that, but AI can predict it. Okay, years in advance for Parkinson also. Okay, so that is a huge thing. Okay, it can do increase automation of routine tasks, which will cut down the workload. Okay, this is used in clinical settings quite a bit. Okay, so what are the remaining challenges which we have not, which you guys have to make sure? Okay, so true deep medicine. So Topol is the one who used the word deep medicine. Remember the first time I showed you in the first slide. So Topol's vision includes a future where AI complements deep phenotyping, comprehensive patient data from genome to lifestyle, but we don't have that much data as yet, which we can add it to the main system. See, your data is not accessible to the healthcare system that I am doing, and yours is not accessible to your healthcare system. So they're sitting in the doctor's office as scribbled notes. It's not part of the whole system, right? So human traits like empathy and creativity, you want AI to be empathic. It is very important to be, when a patient is there, you have to be very empathetic to that person. Okay, because we are humans at the end of the day, we are not robots. So that makes a huge difference. I told you about placebo effect, right? What is placebo? That whenever they do clinical trial, huge percentage of people get sugar pills and they get better. And the people who are getting actual drug, they may not get better. And they always filter out, oh, placebo effect, then this drug is used. If large percentage of people who had sugar pills got better. But why they get better? Because they have this strong belief that they're getting the right drug. So human interaction with the patient is extremely important. If you want to be a doctor, usually people as a surgeon are not empathetic, but that is not a good thing. You have to have a doctor who really cares about you. That is a very important thing. Okay, so many AI works well in research or controls settings, but robust large-scale clinical validation, this is still limited. There is always a diversity issues. Like people who are rich, they get healthcare access, their data are available. For others, they don't go to healthcare, their data is not there. The biggest problem is this, okay, the black box problem. If AI is telling you, so suppose you are a doctor and I'm a patient, I come to you and you go to AI and AI says, okay, give her this medicine. So you don't know why AI is telling that give that medicine. So there's a black box. You need to know why AI is making decision. How can you trust AI to give the thing? Okay, AI is not a human being that you can say, but if it is telling you, it has to explain to you why it is telling you, right? It cannot be black box. You feed in, it's not God that it is telling you this and you will trust it. There's no trust of faith here. You have to know why it is telling you. So the black box, it has to be transparent. It has to tell you exactly why. Okay, so that's the thing, trust one. Always the ethical issues will come up when AI is there. Okay, and obviously it will not maintain the doctor patient thing. Data privacy is a big thing. So we never want under any circumstance that your data should become public. And imagine a situation that worldwide there is one repository where all our data is there. How will you control your privacy there? You have heard of it, right? That somebody in England, so Princess of Wales, she had cancer, but it was not known to us that she had cancer. All we knew that she is unwell and she is not going to do whatever she's supposed to do, right? The worldwide it was there. Then we heard that somebody stole the data because uh, newspapers will pay enormous amount of money for this kind of information. The moment the data was stolen by a nurse there who worked there, immediately Royal Palace announced that she has cancer. And they said why she had to announce cancer. They did not tell which cancer, but they didn't have to tell us that she had cancer. It is her private thing. But why she was, because the data was stolen. So it was going to come to public domain anyway, so they preemptively said that she has cancer. But this should not happen to any of us. All our data in US, nobody can access my data, not even my husband, unless I give the written permission. So when the doctor calls me and they never leave a message on my cell phone, on my voicemail, because they don't know who else may have access to my voicemail. So they will always say, call me back. And if my husband has to talk on my behalf, I have to explicitly say yes. Then they will ask me identification that it is me. So I will give my social security number or something. And then they make sure it is me who is authorizing. Then they will speak to him. So basically the person who is closest to you cannot access your this thing. So it has to be private. Because outside of your own family, nobody knows who is real and who is not real. So unless you are authorizing it, it's okay. But your data, if it is in one public place, wherever it is, how will you maintain privacy? And it is extremely important that you maintain privacy. So the good news is that AI will predict everything, but then you don't want everyone to know about you. Okay. And black box, I already imagine if AI is telling you, give her that, you need to know why. As a doctor, you need to know why AI is prescribing. As a patient, I would like to know why AI is prescribing, both ways. Okay. So here is the thing, the data tsunami is there, but how do we complete this thing? And then there is this thing called NLP. Can anyone tell me what is NLP? It is natural language processing or something like that. It is written in there. Computer reads in a different manner than what doctors write. So everything should be readable by computer in computer language so that they can make sense of it. So there is a tsunami of data, but it is not structured data. So this is silos to data lakes. Silos means my data is in this box, your data is in this box, this box, this box, this box. So we have to make one place where all the data goes, because we have to feed it to AI so that it can learn how to make predictions. It has to know that she got metformin and she had this reaction in the long term. These were the side reactions. That's how AI is built. AI is built on the data that we feed him. it cannot predict anything unless we tell them that 1 million people had COVID drug and this was the reaction in each and every case with this kind of genome. Why few people have reacted adversely to vaccine versus 99% people who had favorable response? You have to first feed the data to AI to make the connection before it can tell you how this medicine will work on other people, right? So that's what it is. Right now it is in silos, and then we have to make a data lake for improved survival. NLP is natural language processing, so you have this so much electronic records, but you have to make sure that it is learned by the artificial intelligence. So natural language processing is a field of artificial intelligence and computer science focusing on enabling computers to understand, interpret, process, and generate human language. Okay, so you have to provide all the data in their own language so that the computer can interpret what it means. Okay. So one day we hope that AI will help. It is already helping us a little bit in surgery, but it will help us more in surgery, robotic surgery. Okay, so finally, what are the future horizons, okay, of AI? So I obviously made this picture using AI. So the first version of this picture did not have any of these things. So I said, annotate it. I'm not able. So it wrote, give it to me in writing that this star means this DNA means this. I said, well, that is not good enough. Just write it in the figure. It's very funny. You can actually converse with that. This is not good. Why don't you write it like that? And then it wrote all the things. What is the meaning of this thing? So basically you can see everything. This is robotic arm that it will help in surgery one day. Okay, this is data analytics displays. This is human doctor in a lab coat, AI medical imaging. Okay, this is the future is very bright. That's what this star shows. Okay, but this is more written here from precision. See, we came to precision thinking precision is so good, right? We were just saying, oh, we are in precision medicine. We are also excited. But the future is not even precision. It's predictive and personalized and preventive, right? So our goal is not to go to precision, to go to preventive. Prevented. Digital twins of patients. Before we are given, they test it virtually. Trust me, you guys will see this in your lifetime. It's not going to be very far away. Possibly we'll see in our lifetime, but definitely you guys will see in your lifetime. This is not going to be expensive. Once it is there for everyone. Okay, AI designed drugs and biologics. Okay, deep medicines means gene and cell therapy, autonomous and augmented healthcare, learning health system. Okay, ethics, convergence of all different kinds of technology. Okay, so this is future. So future deep medicine will not just treat disease, it will continuously model, predict, and prevent it at the level that each human individual. How many of you have seen Star Trek Next Generation? Anyone? No. Anyone has seen Star Trek's series which started in 60s? No. Well, I have only seen the Next Generation. So this is Star Trek. Gene Roddenberry was the one who made it in 60s, and it ran for several, I don't know how long, very popular. Then he made some in 70s. They were not as popular. Then in the 80s, he made Star Trek Next Generation. And I saw it, and I really loved it. Why I loved it? Because it is, I don't know, in 2400 or 2500 or 2700, I can't remember. The reason I loved it, because it was in a time when humanity has risen above this constant fighting, violence, and any disease, they just do this, the whole thing is gone, all this thing. So I loved it, like, anything, huge injury. Okay, anytime you have to go to America, you don't have to physically take a plane. You sit in a beamer, it will beam you out there. And then it will beam you out here. So it's just so nice. You don't have to take plane or something. You can travel any distance, like that kind of thing. So it was a lot of fun. But the best part was there was no war, there was no, and they go out of the way to say that there is no war. We have learned our lessons. So it's all the real good thing. It is like a utopia. Okay, and then they're going to new planets. It's the starships. It's always going to new planets and new adventures kind of thing. So that one was very good. So anytime I see deep medicine, I think of that kind of thing. But then I have to tell you the other part of the story. So it was very, very, very popular, right? Then they made Star Trek movies. I'm not very fond of Star Trek, so I didn't see all others. I just saw the next generation. So recently, I think 4 or 5 years ago, so the hero of that movie was, obviously I'm not remember, Patrick Stewart. He was the main captain. Patrick Stewart, I may be saying it wrong, right? So he was the captain of the ship, and there were others. So they made a new Star Trek recently, 3 or 4 years ago, with Patrick Stewart. Now he's a lot older. That time he was in 50s. Now he's in his 80s. So they made, and there is so much war. It is after that time, so much war. So that ruined the whole thing for me, because we were in a utopia like thing, and again they're fighting. So after 1 or 2 episodes, I gave up. I don't want to see the war and everything else. But my point here is that it is completely foreseeable that anything will be treated without any effort. We don't have to go through huge things there. Okay, so final three slides. Okay, so this was part of your, actually I met a faculty member and he said, I'll come to your last lecture, which he is thinking is after lunch. So he said, I want to hear about your rainbow diet. And I said, rainbow diet? I was thinking to myself, I remember I said that in my Gyan course brochure, but when I made my 30 lectures, rainbow diet was never there. So I went back and checked my notes, and in that, rainbow diet was, so in tutorial, I did not give a specific topic because I'm supposed to ask you questions. So, but only for the 30th one, I had given this title, Disease Prevention, Rainbow Color Diet, Vaccination, Lifestyle Modifications, and Early Screening. So I added it here because it's a very nice place to stop, finish this lecture. So best thing is disease prevention. Don't let the disease come, and this is completely in our hand. So lifestyle modification, healthy diet, physical activity, vaccination, stress and sleep, avoid harmful habits, regular exercise, all of these things. When we do exercise, body knows all the good things to upregulate, body knows all the downs, the bad things. We don't know what are the bad things. Body knows. It will automatically do all the right things. Early screening, I have already told you a lot of stories about early screening, how preventive it is. So reduce disease burden and mortality. Vaccination. Vaccination is really good. Individually and as a society, COVID, see how fast it spread, right? COVID was so bad that I heard of a patient in India when it was just starting. So initially we were laughing about stuff because we never thought it is that serious, right? So we were having a lot of jokes floating around and things like that because, we had never encountered this in our entire life. So I heard of a patient who was in a hospital, COVID thing, right when it was starting. And the patient left the hospital, took a bus to go home, and everybody in the bus got infected. That time it was unheard of, so we were just saying, oh, this is not possible, this is exaggeration, but it is the truth there. Okay, so you are protecting yourself and you are protecting the community by doing that. And global impact, so many diseases have been eradicated, right? Initially, when I was a child, we used to read as a history lesson. I have read, oh, plague used to wipe off the villages and villages. Now these diseases don't exist. We have eradicated diseases because of vaccination. And this is the final slide, the rainbow slide. Okay, so if you eat good food, like red food, of course, tomato, tarbooj, strawberries, all of these things, orange ones, green ones, purple ones, white. Purples are very good, like blueberries, right? They are considered superfood, so rich in antioxidants. In India, you don't get fresh ones, you get frozen ones, I know. Okay, I don't know from where it is coming, if it is coming frozen, but they are extremely good. But we get eggplants, right? Eggplants are bagan, right? We get that. Do we call them eggplants or we call them brinjal? Still brinjals, right? In my time also brinjals. So I didn't know if the vocabulary has changed, because in chicken in my time was not, I'm a vegetarian, but chicken was not called chicken, it was called something else at that time. In my time, I don't know what it was called, but it was not called chicken. In I went to US and it was called chicken. You guys will not know because you guys were not born at that time. But vocabulary changes, okay? So brinjal, we know, lady fingers, right? We call it okra there, okra. Okay, so basically, rainbow diet is the way to prevent disease. Your diet is the one which is playing a vast role. Okay, we finish here. That is the end of this lecture. Wow, we finished it. Okay, you guys can ask me question now. Okay, question time, guys. So let me give you some information first about the exam tomorrow. There are 10 questions, and I have given half a page for each question. Okay, so it's not like one or two or three sentences. You may have to write something there. So one question is like, for example, what are oncolytic viruses? What are the different ways? I have made a question on oncolytic viruses. It was such a big hit. I was going to put a smiley face there, but then I did not. I said, well, the exam, you have to maintain some proper decorum for the exam. So one of the question is on oncolytic virus. What are the different ways by which oncolytic viruses work? So of course, they attack the tumor cells specifically because they replicate in tumor cells, and then they form a bag and they lyse it, and then they activate the immune system. Then you can also add toxin to the wire and it will release it. So different way. So this kind of questions are there. So it's not like one sentence, what is oncolytic virus? There are 2-3 things in the same question. Every question is like that. Okay. In some cases I say, oh, give me an example of that. So I have tried not to become extremely specific about something, but if I ask you about something, I will ask you in more general terms. Okay, so that kind of questions are there. There are 10 questions. Of course, I could not put everything there. Like I cannot. 10 questions are not enough for this whole 30 lecture course. So, but I have. So it's few of your favorite topics may not be there, or few of your favorite topics may be there. I don't know. Okay, so it is like that. So at least read all the important things. You should know the definition and understanding of all this. And in most of the cases, I have said, give me an example to explain that. Explain using an example. So I've tried not to say, okay, give me this example. So whatever it is, if it is oncolytic virus, take one example. If it is immune checkpoint therapy, take one example. If it is PGX, explain using one example. What is PGX? Explain using any one example. So at least have one example in your arsenal. If you're making someone understand, use one example. So that should help you. So pharmacogenomics, what is a nanobody? How does it work? What is a PROTAC? How does it work? This kind of thing. What is an antibody? How does ADC work? versus simple antibody versus ADC, right? CAR T therapy, tandem CAR T therapy, kind of thing. I have specifically not asked you about when I was giving you crash course on immune system because that's very complicated. So I have not asked you anything directly about explain immune system, but I have definitely included questions about the therapy targeted towards how will you target TME. So I have told you many ways of targeting TME, just any one is good enough. I just want to see that you guys got the gist of the course. Okay. All of you are taking the exam tomorrow, right? You guys have attended all the time. Venkatesh has already said that he gets too many emails, I want to attend the course, but the students are not showing regularly, so he said they are not going to get the certificate. Okay, guys, ask me questions.
Ma'am, we have immunotherapy, then we have cell therapy. So in immunotherapy, we are using CAR T cells, engineered.
Please, वो कहाँ गया वो? In immunotherapy, so tell me what are the different things you can use in immunotherapy? All of you give me one immunotherapy thing. Yes. Immune checkpoint therapy, very good, like PD-L1, PD-1, very good. And CTLA4, very good. आपका हो गया checkpoint, मतलब correct.
We can use oncolytic viruses or cancer vaccines.
Very good. That is also immune therapy, cancer vaccines and oncology, CAR T therapy, immune system. और कोई भी है क्या? क्या? Antibody drug conjugate, very good.
That is what I'm T cell specific antibody. Bispecific antibodies.
You can use bispecific antibodies, very good. You can use bispecific antibodies, yeah. You can use a PROTAC which will bind to a nanobody, which will bind to a tumor cell, and you can bring the drug there also. Very good. So go ahead. So what was your question?
I wanted to know that what is the difference between a CAR T cell therapy or immunotherapy, specifically CAR T cell, and cellular therapy, because there also we are engineering the cells.
So CAR T cell therapy is a cell therapy because CAR T is part of a cell. But if you are giving, when you say cell therapy, that means you are giving a cell. So CAR T or CAR NK or CAR M, they are all cell therapies, right? Because you are giving a cell. But if you're giving a gene kind of thing to turn on something, that is not cell therapy, that is gene therapy. And what was the other thing you were saying? Immunotherapy, if you are giving PD-L1 antibody, so that's not a cell therapy. That is, you are giving just the antibody there. So it is immunotherapy, but it is not necessarily a cell therapy. But CAR T is a cell therapy, but it comes under immunotherapy. Yeah.
And then stem cell therapy can be included under cellular therapy.
Depends on that. Stem cell is a cell therapy, so it is a cell therapy. Stem cell is always a cell therapy. So you should know what are stem cells. I forgot all about stem cells, the pluripotent, the totipotent, the multipotent, the mesenchymal, the hematopoietic, and I definitely have a stem cell question. So what is a stem cell? How will you define a stem cell?
Self-renewal in the places of
So you have to also say the stem cells can make more stem cells. Don't forget that part, because you don't want stem cells to always only differentiate. Otherwise, you will get, how will you keep the, So stem cells can make more stem cells, and then they can differentiate into many, many different kinds of cells. Yes. So that's it. So those are stem cells. And give me an example where you can use stem cell as a therapeutic agent.
in case of chronic inflammation, in order to avoid its like advancement into cancer, you will regenerate cell by giving stem cell therapy.
Tell me exactly how will you give stem cell therapy to prevent cancer from occurring?
In case of, I guess, in, in
Oh,
case of
oh, yeah, she's giving you that. Please take that. All of you remember to take that.
in case of IBDs or any like gastrointestinal inflammation, we can give mesenchymal cell to.
Very good. I had forgotten. Mesenchymal stem cells have immunomodulatory properties. Very good. I had forgotten. I was thinking myself, how can you stop inflammation? Yes, that's a very good one. Very good one. So see, you have chosen any. So you should have one example of therapeutic, whichever you like. Okay, one example of whatever. I had given you regeneration. You can generate the whole breast by that thing. Then bone you can regenerate, right? If somebody, you can generate, you can have a cardiac patch, right? So things like that. So go through that, but just remember one part of everything. It's used as therapeutic, it's used as regenerative tool, it's used as, yes, you can deliver the drug that way. Very good. So you can say, okay, I want to deliver the drug. I will use mesenchymal stem cells for homing property. You are done. Perfect. So you get to choose what you love the most there. That's how the questions are designed, as I recall. That's very good actually. Mesenchymals are really good ones. Then people who get leukemia, they get irradiation because their red blood cells are destroyed. So red blood cells are given from siblings with matching HLAs, right? So that is also use of stem cell therapy, right? You get new bone marrows. I don't know about this thing, but I've heard that taking out the bone marrow from a healthy person is very painful, and to give it, but people, but it is better than kidney donation, less painful than kidney donation. What is a HLA? Leukocyte antigen. But what is it? MSC equivalents. So HLA1, HLA class 1, HLA class 2. Great. I love our discussions so much. That's why I've included some things here and there as I learn about it. And liquid biopsy I was saying, oh wow, you can do liquid biopsy. That's really awesome. Because we don't want to open up the patient. So doctors are really against opening up someone because anesthesia has a serious side effect. I did not realize it. Anesthesia is so dangerous.
There is only one stage if you cross the patient will have paradoximal something. I don't remember it's stage of sleep cycle.
Okay, I was thinking doctors are overreacting. I never realized anesthesia is so dangerous. Like, anytime they put you to sleep, they always make you sign some paper that you may never wake up from that. So that is very scary. Exactly, exactly. But the good news is that it is shared in the bloodstream, and blood is least invasive, you know. I mean, taking out blood is a lot easier than anything else. It's just a prick of a needle that you have to suffer. Yeah.
So then we can do it with local anesthesia, it's okay.
Yeah.
When it comes to generalized,
That is very bad. That is very bad.
Inhalation forms like inhalers
Oh,
kind
you
of
do
form
use it all the time. You
for
use
lung
all the
cancer to
time.
be
Actually, it
specific.
is written in one of the things there. So many drugs that you have to send it to the brain, they do not cross blood-brain barrier. But if you make it in an inhaler form, it will directly go to the brain. So that's a very easy way to get it to the brain, if you can make it in that way. So one of my friend was working in a company. Its name was something nasal. It was in Arizona, and it used to make many drugs which you could inhale, and it will directly go to the brain. So that's a very good idea, and it has been realized. See, keep in mind that you guys are very young and you're just learning about it. So for you, it's a brand new idea. But if it has been realized, then you should really pat yourself that you thought of the right thing, right? So your thinking is in the right direction, which is a big thing. Okay, so that's very good point. And also, you guys should be very cautious of all the things that you put it on your skin because it is directly going to your bloodstream. So we are very careful of what we put in our mouth, but you have to be equally careful of what you put in your skin and what are the things you're holding with your hands. Who was telling me about the fertilizers? Who was telling me? You were telling me about the fertilizers. See by hand how bad it is, right? Because it is directly going in the bloodstream. I talked to so many people during lunchtime and, you know, evening and this and that. So I get confused who was getting, because I'm getting too much input of, information from all of you too. So I'm confused at the end of the day where it is coming from. You guys are living, at least as students, you are living in a nice place with a lot of greenery. So that is a blessing if you get a job also in a clean, nice place where you are not exposed to dust. Go ahead, next question. We can even have 1 hour tutorial at the end of the day. We can have as long as you guys want. We'll just have as long as we want and we'll stop when we are done with it. Okay. We can have 2 to 3, 3:30. Okay. Next question. Today Vance is not there. Just take that thing, mic.
In the slide about CRISPR, like we were studying CRISPR-Cas9, there was a point which said editing the epigenome. So was that just using the targeting mechanism of the Cas to do the targeted demethylation and all that stuff, or was it something different?
Uh, if it is targeting the epigenome, you want to fix the enzyme which is doing that. Because if you just remove the methyl group or something, that is
Okay.
post-translational modification.
yeah.
CRISPR cannot do that.
Yes, ma'am.
CRISPR can do it at the DNA stage only, right?
Hmm.
So it will actually target the epigenetic enzyme to have a long-lasting impact.
In CRISPR, if we are removing one particular gene in between the sequence, and then it will get like joined with any mechanism of DNA damage repair, then that new gene will be created at that site. Then then it may have its own like other
So
expression.
before you use CRISPR, the problem is already there, right? Before you use CRISPR. So you can remove the whole piece of there. So if it is BCR able, you can cut down the able BCR part, or you can remove the whole BCR able. If something happens afterwards, if you have removed the gene and some other part is going, it may not have any impact because the gene is missing. So what is that other part going to control there? But if it originates because you have done CRISPR, then you can go back and do the CRISPR again to remove it. So CRISPR can change the DNA sequence. CRISPR can remove the gene. CRISPR can add the gene. It can do a lot of things. So the other Gyan course that I taught actually about CRISPR also. But I kept CRISPR actually technology out of this because most of you already know how CRISPR works. So, and that is not directly related to this thing. Exactly. It's all in the primers. the cgRNA that you create, you can exactly tell what exactly to do. It's a piece of cake to do it, actually. Our collaborator, he created, now CRISPR vectors are available in AdGene, but that time they were not. So our collaborator created his student created a CRISPR vector, and we used it in our lab. And so they are all part of that paper, and it is just very easy to change any gene. It's very, very easy now. CRISPR is like everyday thing now. It's not a big thing. But it was a huge discovery. And we got the inspiration from bacteria. So bacteria are the one who have this CRISPR mechanism, and that is there to protect them from foreign attack. And that's what the scientists adopted. So Mother Nature has all the solutions. It's up to us to see the solution. Many times we see that if you are a Superman, you always find something which most of the people do not see. Superman does not mean like the superhuman doesn't mean just the strength. It means the smartness, right? So what this AI is doing, that when you show the pathology report, right, every pixel, it can read every pixel. It can see so many things in each pixel and do so many correlation compared to what we are seeing. So that's how it can see a lot more than what human brain and eyes can comprehend. Yes. Yes, it is. Please give that. Just 1 second. Repeat your question.
So, retinoblastoma protein, you told us it is a tumor suppressor and its indeficiency causes some tumors. So, ma'am, are there any specific protein? Are there any specific tumors which are caused by its indeficiency, like BRCA1, BRCA2 deficiency causes more breast cancers and ovarian cancer than other cancers? So, is there any specific cancer to
Yes,
this too?
absolutely. So retinoblastoma proteins are also known to be deleted in certain kinds of cancer. So when I was telling you about that, it actually specified which cancers it is specifically deleted. So when you do the cell cycle here, right? So when you're going through G1, there is a point where retinoblastoma protein is very important. So that's when it controls whether it should go past that point or not. So retinoblastoma protein is involved in cell cycle, and it is deleted or mutated in certain cancers, but overall a large percentage of cancer. Then there is another, P53 is deleted. There is another protein called P10 that is deleted. That is also a tumor suppressor. Then CDKN2A, remember, the MTAP one is a passenger which gets deleted by mistake because it is just next to it, like a bystander which gets deleted. So yeah, so tumor cells go out of the way to delete these proteins. So imagine how smart they are, like how smart they are. Somebody had Alzheimer's disease long time ago. So there was some kind of viral infection which led to Alzheimer's, and at that time it was not known that viral infection can lead to Alzheimer's disease long time ago. But the thing is that they grow, and they kill the patient. So ultimately, they are live cells, but they all die. Same thing with cancer. It is taking so many different pathways to survive, but you kill the host, the cancer also dies with the host. But the thing is that it is what it is. Prevention is the best medicine. That's the message here. So let me ask you, if you have to summarize the course, I will ask you guys, what are the things that you learned? And it is personal to all of you, right? So you tell me, what are the things you remember that you learned in this course which made sense to you? Like, I attend a course and I have some take-home message. I attend a lot of courses which are different from these courses, right? And then somebody said, okay, what did you learn in this course? And if I have to say it in one or two sentences, it's different from everyone else. So what are the things that you learned that you didn't know before? I'm going to go around and ask everyone. CAR T cell therapy. So those are the things you really learned about it. What about you? Almost everything was new to you. Yeah. Do you remember something like you have, obviously you cannot remember the details, especially for these kind of crash courses, but what are the things which resonated most with stem cells? Very good. What about you?
Ma'am, before joining this course, I had a little bit of idea of everything, but I was not having the clarity that which one to prefer, why to prefer, where to prefer. So now it's a clear picture
Of
everything.
all the things. And then the thing is that you have the notes. You can always go back and read the notes. Okay, so that's the most important thing. That is your thing. It is all in one place. What about you?
Pharmacogenomics was a new concept, and those are learning.
These are very new for you. Actually, before I prepared this class, I did not know about pharmacogenomics myself because that has not crossed my research thing. And I was saying, oh, now it makes sense why I did not respond to that drug. So it is very strange. I have a chronic headache problem, and one time I was given a medicine for chronic headache, and that medicine was okay, but I didn't realize that I was very quiet when I was taking that medicine, and somebody close to me said, you are always very depressed. Not depressed, matlab, down, not energetic. And that medicine is not supposed to be, it's antidepressant side effect, but I was depressed, and I did not know that, and somebody commented that. And then one day I actually forgot to take the medicine, and I was just so chirpy that day. All day I had a lot of energy, then I never took it again. So its side effect was antidepressant, and I was taking for headache. My doctor was horrible. She was prescribing all these stupid medicines to me, but I got depressed with that. So my pharmacogenomics is different, and I did not know about pharmacogenomics till I prepared for this class. So that was my learning too, pharmacogenomics. Yes, go ahead.
Ma'am, basically, I have a major in immunology. So, I mean, yeah, it's core of the discipline in which I am interested. So,
Oh, please give that. It has, yeah. You have a major in immunology.
yeah. So,
Come from that immunology background, all of the things which I can relate to made it easy for me to learn more of the concepts. Also, the questions I've asked is something which it's basically my imagination coming to the reality and getting the real time answers and resolving it. It's a new thing for me and it's really amazing.
Thank you, thank you, thank you. Your turn. Oh, yeah, you can go ahead. You have the power. Just 5 minute में आ रहे हैं, हाँ 5 minute में आ रहे हैं.
So I'm also interested in immunology, and I've always looked at disease proliferation and been very interested in the mechanisms from the side of the disease. But in this course, drug delivery and like the methods of approach to the disease have been so fascinating to me that I think I'm more interested in that part now.
That is very
Because I
good.
used to think from the side of the disease now I'm thinking from the side of the human being.
Exactly. That's very good. Go ahead.
Ma'am, pharmacogenomics was a new concept and precision medicine.
Precision medicine. Of course, we did not know actually what is the meaning. I did not know actually what's the real difference between personalized and precision. Precision seems more precise. So I thought, you go from personalized to precision. I learned no, you go from precision to personalized. That was a wake up call for me too. Yes, go ahead, Vanshika.
Before I only knew that conventional therapies of cancer, but I got to know immunotherapy, which was very interesting for me, and it was very fun process learning all this. And as he said, it was very fascinating to to know so much new things that we can approach towards cancer.
Yeah.
Before I used to think that it is a uncurable disease, like we cannot
It
cure
is
it,
not.
and
It is
so I
not.
got to know many new things.
Yeah, you got to, and you guys are going to contribute to many new things, so it will become completely curable. Go ahead.
My answer is kind of similar to hers. Uh, Before the course, I only knew that cancer can be treated, or not even treated is not right word, but the response to cancer can only be chemotherapy or surgery. But now I know how many different aspects to it. I got to know there are different types of tumors and they should be treated differently.
That is also
Epigenetics was a great uh, uh,
introduction to
great
that.
introduction to that. It was very
Yeah,
fascinating thing.
yeah.
That same genetics but different expressions.
Different expressions. That's very good. That's very good. Yes, yes, yes.
And about the cancer and immune cells interaction, that how
Oh, that
smartly
is.
cancer controls immune cells.
Oh,
That was
that
very
was a very
fascinating.
new thing too, immunosuppressive environment.
Immunosuppressive.
Yeah, that is very good. Thank you. Go ahead.
Ma'am, for except the AI part and drug repurposing and nanoparticle part, everything was new for me because I am currently working in both all the three topics, and I have also worked in the nanoparticles. I have assisted someone making transdermal patches. So for me, the cancer part was like new. Everything was new for
So
starting
you
from
are
the
in
therapy.
which lab?
Ma'am, in LP University.
Which university?
LP University, lovely
LP
profession.
University. You have worked on nanoparticles.
Yeah,
and
there I have
everything.
assisted someone.
That is very good.
Yes,
That
ma'am.
is very good. But now you have a more complete
Yes.
picture there. So the bottom line, the last message is there is that it is just constantly evolving. And I've not even covered all the topics. I've covered, whatever I thought were important, but it is much, much, much beyond that. But at least you have enough background that you know what questions to ask ChatGPT, right? Because you have to also know how to use it, right? Because it is just so that is very good. All right, guys, let's have lunch and then we will meet again at 2 o'clock. Okay. Bad you hear all about people, but then at least you know about because you're overwhelmed hearing about how smart it is, right? So bus ka dीजिएगा. One more thing I came to know that I was like wondering that how we detect like it is cancer. We do PET scan, but now I understand the basic reason behind PET scan is dysregulated metabolism. Exactly. That was the simplest answer. Simplest answer why we do, and now you know you don't even have to do PET scan. So I am also learning with you guys, right? I saw that, yeah, blood screen. Wow, wow, wow. This is so exciting. We don't have to open up the patient, right? So I'm also learning with you because I'm more on the research side. I'm not on the doctor's side, right? So I got to learn a lot of things too. All right, चलते हैं फिर लौट के आते हैं फिर और करते हैं. Any targets?
Initially, it was made for one specific target, but accidentally it worked.
It was an accident pharmacology for Gleevec. That's when they realized polypharmacology is very good. Acha, what is deep medicine? Precision medicine plus AI, not that. Very good. Acha, how will you define CAR T therapy? Anyone?
Enabling T cells with CAR receptors so that they can target the tumor cells, recognize tumor cells while not attacking the normal body cells and not get attacked by the body's immune cells.
That is very good, but would you like to add something more to it?
It's sort of a platform which brings antibody directly in close vicinities with the target.
It has nothing to do with antibody. CAR.
Genetically engineered T cells.
These are genetically engineered T cells which have SCFP receptor, so nothing to do with the antibody. Just be cautious when you're writing the answer. Don't be that generic because it would be wrong. So you have SCFV receptor that binds. See, just be very accurate, Venkatesh. So it will recognize tumor cells by binding some tumor antigen. Okay, so make sure you say tumor antigen, and then once it binds, then it has the co-stimulatory molecules and other things, so it will spray. It is not going to bring antibody there. It is it is weaponized, so it will spray grand enzyme, perforin, and all those things. So it will spray and kill it. Okay, so it doesn't depend on something else coming and killing it. It will just spray it. So make sure be cautious in answering that. So look at it. Okay, so CAR T, polypharmacology. So tell me 3 or 4 different ways of how will you do active targeting. Tell me different ways of doing active targeting to tumor cells. Huh? Very good. So that's one. Second, again some specific target, but nanobody coupled to something, right? Coupled to a drug. Very good. And that so make sure you write nanobody raised against a specific tumor antigen, tumor specific antigen. So write it properly. Okay, you may know it, but I have to know that you know it completely, right? So that is okay. What is a PROTAC molecule?
It links between gene of interest and to the protein of interest.
Protein of interest, not gene of interest. Gene is DNA. Protein of interest on one end. So write it properly. So it binds to the ligand. It has a small molecule which binds to the protein of interest. Write it properly. And that protein of interest, the linker is there, and it is not E3 ubiquitin. That would be wrong. E3 ubiquitin ligase. That is a ligase. E3 ubiquitin is no word. E3 ubiquitin ligase. Okay, so that is there, and it will bring it. So how is it superior, or how is it advantageous to use a PROTAC as compared to small molecule? And they degrade it. So you are also taking care of protein-protein interaction with that one, right? So okay, that's very good. और क्या-क्या चीज़ हम लोगों ने पढ़ी है? Give me the topics and I'll ask you.
G protein.
G proteins, right? So how do you define a G protein? Define a G protein.
Intracellular signaling protein.
Define G proteins in 2 sentences. आप लोग जो है ना गलत कर रहे हैं. हम अगर बहुत लिबरली भी पॉइंट्स देंगे तो भी नहीं दे पाएंगे इस केस में. तो आपको एक्यूरेटली बोलना है. देखो, वी आर साइंटिस्ट, वी हैव टू आंसर वेरी एक्यूरेटली. सो व्हाट आर G proteins? How do they get the name G proteins? आपको ट्राई मारना है. G proteins are those proteins which bind either GTP or GDP, and when they are bound to GDP they are turned off, and when they are bound to GTP they are turned on. So G proteins are, this is the definition. G proteins are molecular switches which bind to either GTP or GDP. When they are bound to GTP they are turned on, when they are bound to GDP they are turned off. That's the definition. So G proteins means they have to be bound to either GTP or GDP. So don't say second messengers or anything. That comes a lot later. That is not a definition of G protein. G proteins are GTP or GDP binding proteins, and you have to specify GTP on, GDP off. Okay, and you also should say it's a molecular switch. Okay, so that's very important. That was a good question. What do kinases do? Phosphorylate by using? What is the difference between kinase and a phosphorylase?
Phosphorylase are inorganic phosphorylase.
Very good. They are not using ATP. Good. That's the difference between phosphorylase and phosphate. And how does CTLA4 works? What is happening in that immunotherapy? That is the hard one. PD1, PDL1 is easy. Explain to me PD1, PDL1 first. Anyone?
PD1, PDL1 are the normal immune checkpoints where PD1 binds with PD1 of the T cells binds with PDL1 of antigen presenting cells, and it turns it off, like if it is hyperactive. But some tumor cells, as defense mechanism, express PDL1 and interacts with T cells PD1, turning them off.
Very good. It is perfect answer. 100% correct. बहुत अच्छा. हाँ. So you know CTL4A. How does it work? That is a complicated one. If you can remember.
Yes, there are three signals.
There are three signals. Acha, I can give you a hint here. I have not asked you about CTLA4 because I thought वो थोड़ा hard हो जाएगा, बहुत कुछ याद रखना होगा, पर आप मुझे बताइए, at least आज बता दीजिए.
CTLA4 is expressed in T cell when they are hyperactive. The tracks have B7 receptors which binds with PTLA4. Sorry,
CTLA4.
CTLA4, turning down the activity of T cells.
So this is very good. So what happens is that under normal circumstances, T cells, after they get activated, then they will express CTLA4, and then APCs are the one which will have the B7, which will bind to CTLA4, right, and turn it off. But in tumor cells, they will recruit Treg cells, which will bind there and turn the signal off. So this is totally correct. So what you said is totally correct. Okay, so CTLA4. So these are immune checkpoints. So what are immune checkpoints? You can explain using PDL1, PD1, right? So what is synthetic lethality? Explain using one example, any one example of your choice. Yeah.
Like synthetic lethality, one gene is not working, and we can block the other gene, so the overall cell dies.
So why is it called synthetic lethality then? You have to explain. Don't take the shortcut. Explain. Go ahead.
Because in cancer progression, one gene is already defected, and we are targeting another gene. We are causing lethality.
By that, but normal cells have both genes there, so if you inhibit it, it's not going to affect. So explain everything. Okay, तो थोड़ा सा ये बता दीजिएगा कि normal cells में both pathways A and B are there, so if you inhibit A, B takes over, if you inhibit B, A takes over. But in tumor cells, one pathway is already defective, so if you hit the second one, cells die. That is synthetic lethality. Correct? और कौन कौन से टॉपिक्स हम लोगों ने कवर किया था? हाँ, थेरेपीज एंड हॉलमार्क्स. हॉलमार्क्स तो पूरे समय बोलते ही रहे हम लोग, है ना? I can tell you I don't have any question on microbiome. तो थोड़ा सा रिलैक्स कर सकते हैं आप लोग. क्या? Yeah, so I want to ask you genetic and sporadic diseases, what does it mean actually?
Genetic diseases are inherited genetically from the
Germ cells.
from germ cells, and the sporadic diseases are the ones that we acquire during the
Exactly, that's what it is. Okay, that is what it is. And most of the diseases are sporadic. And what is exposome?
Expression could be any external factor which influence the kilometers.
Exactly. And so sporadic diseases are caused by exposome. Okay. आप लोग के लिए ना लाइफ लॉन्ग लेसन है ये कि टेक केयर ऑफ योर एनवायरनमेंट. Nobody in the world, even the richest person जो कि बहुत ही busy होता है, nobody is busy enough not to take care of yourself. You can just always have to take care of yourself, because life is it's very strange. It seems like अभी तो शुरू हुई है और फिर बहुत जल्दी ख़त्म भी हो जाती है ना. तो मतलब देखिए, जितने एज तक रहना है जिसको, वन हैज़ टू बी कम्प्लीटली हेल्दी, राइट. You cannot be a walking matlab unhealthy person. That's what I'm saying. So the take home message from any such course is that really take care of yourself. It's not a joke. Next thing, I like that you do the exercise thing, body. That is very good to hear. I'm really happy to hear that thing. People are much more jagruk these days compared to in our time. People were never doing exercises there at that time. People didn't know, but at that time there was no computer, so people were always walking around doing the job. So exercises ho hi jata tha sabka. There were not so many cars. It was culture that you walk to close by places. It was not considered social symbol or something, because it was the habit, so we used to get the right amount of exercises anyway. सब लोग ग्रुप बना के स्कूल जा रहे हैं, तो it used to be fun walking, even going to school, so, and things were safe and everything, और फिर we used to play, मतलब इतना physical activity, because we were not playing on computer, we were just actually playing. और इतना अधिक खेलते थे ना कि वो सारी एक्सरसाइज बिल्कुल हो जाती थी. And we were just climbing up and down stories, ऊपर नीचे ऊपर नीचे ऊपर नीचे ऊपर नीचे, पूरा दिन दौड़ते. So, and that is why now exercise is important, because in IIT Kanpur we didn't have electric vehicles. We had to walk or bicycle से जाना होता था, मतलब साइकिल से, जोर से. हाँ, biomarkers, it's very important to remember all the biomarkers. You do have a question on biomarkers, so you should remember all sorts of biomarkers. That's a very important one. I forgot.
Drug resistance, therapy resistance.
Yeah, read about therapy resistance also, biomarkers also. I did not know that you guys are going to get the grades also, because I was thinking it is pass and fail, so I was relaxed कि सब लोग तो पास हो ही जाएंगे. But I would not like to give you a bad grade, so please do well. Okay, I would feel very bad किसी को B देना पड़ा तो, है ना. I'm going to be as generous I can, but I still have something that if you write something wrong, I cannot make it right. I have to respect that thing. I'm too old-fashioned for that. Okay, so please do very well. Ask me as many questions you want to ask me today.
Do you have something with cancer vaccine?
जोर से, जोर से.
No. I'm asking that question. Do you have something in question, cancer vaccine also related to?
Could be. I don't remember, honestly, I don't remember. Usually what I do is that I will tell you one or two things which is not there, like microbiome, but I don't even remember. I'm not so fond of microbiome. I mean, it is extremely important, but not an important topic I don't know what to ask you about microbiome. That's the thing. Epigenetic gene therapy is very important. Know about all the different mechanisms. Tell me epigenetic mechanism. I know, Venkatesh, you are very good in epigenetics. I have noticed that. And you get it very quickly. You propose a lot of things epigenetically. So tell me.
If two persons have similar genetic makeup for the same subject, but they have different expressions of protein, it might be because of their epigenetics. Epigenetics is the expression of the same genomic material because of the condensation pattern of the genetic material of the DNA.
Yeah,
The
so
tightening and loosing of the DNA
due to which epigenetic modifications. So make sure you say methylation of DNA, which is acetylation. Methylation closes it. So here is the thing between you and me, right? So we may have the same epigenetic mechanism, but within us also, depending on our environmental conditions, epigenetics can change. So for example, um, because of stress, epigenetic pattern may change. So it is not that it is different between different people. It has nothing to do with it. It is all within us that at different times we respond to different things. So I attended a neuroscience meeting maybe 10, 15 years ago. So there the new finding was, which is now a very old finding, that how do you make short-term memory versus long-term memory? Can anyone tell me? Some memories are long-term, right? हमेशा याद रहती हैं, and some memories are short-term. So people who have Alzheimer's, they retain long-term memory, but they don't retain short-term memory. They will forget अभी 2 मिनट पहले क्या हुआ था या कल क्या हुआ था, but they will remember their kids. So what happens in the brain? You probably don't know. It is epigenetic modification. Those memories which are stored somehow, the proteins we store are methylated, or they are differently epigenetically modified, and those memories become long term because of the epigenetic modification, and short terms are not. By the way, robotics in AI, deep medicine can also help. Like you think of something and the robotic arm will move. So जिनके prosthetic limbs होते हैं, तो उसमें खाली सोचने से वो लिम्स को आप मूव कर सकते हैं. जैसे हम लोग सोचते हैं और सब कुछ कर लेते हैं, है ना? तो उसमें खाली थॉट से यू कैन मूव इट. So that is where AI is going.
So do they integrate PNS with prosthetic arm?
I don't know how they do it, but it is thought controlled.
PN is peripheral.
I think somehow they do it. I don't know. I have no idea. I was reading about it this morning actually when I was talking to my niece. So, because she's very much into neuroscience. So, and how thoughts are arranged in the brain in the 3 dimension, and how do we comprehend 3 dimensional space. इसको बंद कर सकते हैं? अब हो गया बहुत. हाँ, अब आपका काम खत्म, छुट्टी. थैंक यू. इतने दिन तक आपने किया, सो थैंक यू सो मच. अब हम लोग बात कर सकते हैं फ्रीली. हाँ, अल्जाइमर स्टोरी. हाँ, अब हम आपको बहुत सारा फ्रीली बात कर सकते हैं, क्योंकि ना हमने वेंकटेश से ऑलरेडी कहा है कि आपको बहुत एडिटिंग करना होगा, क्योंकि हम अपनी काफ़ी